10. Photosynthesis. Figure 10.1. Sunlight shines through a tree canopy. Each leaf of this tree is harvesting energy from sunlight and using it to convert CO2 and H2O into chemical energy stored in sugar and other organic molecules. The tree uses these sugars for energy and to build its own trunk, branches, and leaves. Remarkably, enough sugars are left over to feed other organisms, like moth larvae, shown below, that cannot carry out this extraordinary transformation. How do photosynthetic cells use light to change carbon dioxide and water into organic molecules and oxygen? 10.1 to 1 full alternative text. Key concepts. 10.1 Photosynthesis feeds the biosphere. Rep. 188. 10.2 Photosynthesis converts light energy to the chemical energy of food. 189. 10.3 The light reactions convert solar energy to the chemical energy of ADP and NADPHP. 192. 10.4 The Calvin cycle uses the chemical energy of ADP and NADP to reduce to sugar. 201. 10.5 Alternative mechanisms of carbon fixation have evolved in hot, arid climate. 203. 10.6 Photosynthesis is essential for life on Earth, a review. 206. Study tip. Make a visual study guide, draw a cell with a large chloroplast, labeling the parts of the chloroplast. As you go through the chapter, add key reactions for each stage of photosynthesis, linking the stages together. Label the carbon molecule, S, with the most energy and the carbon molecule, S, with the least energy. A drawing of a chloroplast inside a plant cell. Go to Mastering Biology. For students, in e-text and study area. Get ready for Chapter 10. Bioflix registered animation, photosynthesis. Figure 10.13 Walkthrough, how linear electron flow during the light reactions generates ADP and NADP. For instructors to assign, in item library. Activity, the light reactions. Bioflix registered tutorial, photosynthesis. Ready to go teaching module, in instructor resources. The light reactions. Concept 10.3. Concept 10.1 Photosynthesis feeds the biosphere. The conversion process that transforms the energy of sunlight into chemical energy stored in sugars and other organic molecules is called photosynthesis. Let's begin by placing photosynthesis in its ecological context. Photosynthesis nourishes almost the entire living world directly or indirectly. An organism acquires the organic compounds it uses for energy and carbon skeletons by one of two major modes, autotrophic nutrition or heterotrophic nutrition. Autotrophs are self-feeders, auto means self, and trophos means feeder, they sustain themselves without eating anything derived from other living beings. Autotrophs produce their organic molecules from CO2 and other inorganic raw materials obtained from the environment. They are the ultimate sources of organic compounds for all non-autotrophic organisms, and for this reason, biologists refer to autotrophs as the producers of the biosphere. Almost all plants are autotrophs, the only nutrients they require are water and minerals from the soil and from the air. Specifically, plants are photoautotrophs, organisms that use light as a source of energy to synthesize organic substances. Figure 10.1. Photosynthesis also occurs in algae, certain other protists, and some prokaryotes. Figure 10.2. In this chapter, we will touch on these other groups in passing, but our emphasis will be on plants. Variations in autotrophic nutrition that occur in prokaryotes and algae will be described in Concept 27.3. Figure 10.2 Photoautotrophs. Photoautotrophs, including A. Plants, B. Multicellular alga, C. Unicellular eukaryotes, size 30 micrometers, D. Cyanobacteria, size 100 micrometers, and E. Purple sulfur bacteria, size 4 to 5 micrometers. These organisms use light energy to drive the synthesis of organic molecules from carbon dioxide and, in most cases, water. They feed themselves and the entire living world. On land, A. Plants are the predominant producers of food. In aquatic environments, photoautotrophs include unicellular and b. multicellular algae, such as the scalp, c. some non-algal unicellular protists, such as Euglena, d. the prokaryotes called cyanobacteria, and e. other photosynthetic prokaryotes, such as these purple sulfur bacteria, which produce sulfur, the yellow globules within the cells, c. e. LMS. Heterotrophs are unable to make their own food, they live on compounds produced by other organisms. Hetero means other. Heterotrophs are the biosphere's consumers. The most obvious other feeding occurs when an animal eats plants or other organisms, but heterotrophic nutrition may be more subtle. Some heterotrophs consume the remains of other organisms and organic litter such as feces, and fallen leaves. These types of heterotrophs are known as decomposers. Most fungi and many types of prokaryotes get their nourishment this way. Earth's supply of fossil fuels was formed from remains of organisms that died hundreds of millions of years ago. In a sense, then, fossil fuels represent stores of the sun's energy from the distant past. Almost all heterotrophs, including humans, are completely dependent, either directly or indirectly, on photoautotrophs for food and also for oxygen, a byproduct of photosynthesis. Mastering biology. Bioflix registered animation, the flow of carbon atoms in producers, consumers, and decomposers. Concept check 10.1. Why are heterotrophs dependent on autotrophs? What if fossil fuels are being depleted faster than they are replenished? Therefore, researchers are developing ways to produce biodiesel from the products of photosynthetic algae. They have proposed placing containers of these algae near industrial plants or highly congested city streets. Considering the process of photosynthesis, how does this arrangement make sense? For suggested answers, see Appendix A. Concept 10.2 Photosynthesis converts light energy to the chemical energy of food. The remarkable ability of an organism to harness light energy and use it to drive the synthesis of organic compounds emerges from structural organization in the cell. Photosynthetic enzymes and other molecules are grouped together as specialized molecular complexes in a biological membrane, enabling the necessary series of chemical reactions to be carried out efficiently. The process of photosynthesis most likely originated in a group of bacteria that had enfolded regions of the plasma membrane containing clusters of such molecules. In existing photosynthetic bacteria, enfolded photosynthetic membranes function similarly to the internal membranes of the chloroplast, the eukaryotic organelle that absorbs energy from sunlight and uses it to drive the synthesis of organic compounds from carbon dioxide, CO2, and water, H2O. According to what has come to be known as the endosymbiont theory, the original chloroplast was a photosynthetic prokaryote that lived inside an ancestor of eukaryotic cells. You learned about this theory in Concept 6.5, and it will be described more fully in Concept 25.3. Chloroplasts are present in a variety of photosynthesizing organisms, see some examples in Figure 10.2. In this chapter we focus on chloroplasts in plants, while mentioning prokaryotes from time to time. Chloroplasts, the sites of photosynthesis in plants. All green parts of a plant, including green stems and unripened fruit, have chloroplasts, but the leaves are the major sites of photosynthesis in most plants. Figure 10.3. There are about 500,000 chloroplasts in a chunk of leaf with a top surface area of 1. Chloroplasts are found mainly in the cells of the mesophyll, the tissue in the interior of the leaf. Enters the leaf, and exits, by way of microscopic pores called stomata, singular, stoma, from the Greek, meaning mouth. Water absorbed by the roots is delivered to the leaves in veins. Leaves also use veins to export sugar to roots and other non-photosynthetic parts of the plant. Figure 10.3 Zooming in on the location of photosynthesis in a plant. Zooming in on the location of photosynthesis in a plant. Leaves are the major organs of photosynthesis in plants. These images take you into a leaf, then into a cell, and finally into a chloroplast, the organelle where photosynthesis occurs, middle, LM, bottom, TEM. Figure 10.3 Full alternative text. Mastering biology. Video, chloroplasts in motion. A typical mesophyll cell has about 30 to 40 chloroplasts, each measuring about pi. A chloroplast has two membranes surrounding a dense fluid called the stroma. Suspended within the stroma is a third membrane system, made up of sacs called thylakoids, which segregates the stroma from the thylakoid space inside these sacs. In some places, thylakoid sacs are stacked in columns called grana, singular, granum. Chlorophyll, the green pigment that gives leaves their color, resides in the thylakoid membranes of the chloroplast. The internal photosynthetic membranes of some prokaryotes are also called thylakoid membranes, see Figure 27.8b. It is the light energy absorbed by chlorophyll that drives the synthesis of organic molecules in the chloroplast. Now that we have looked at the sites of photosynthesis in plants, we are ready to look more closely at the process itself. Tracking atoms through photosynthesis. Scientists have tried for centuries to piece together the process by which plants make food. Although some of the steps are still not completely understood, the overall photosynthetic equation has been known since the 1800s. In the presence of light, the green parts of plants produce organic compounds, and from end, we can summarize the complex series of chemical reactions in photosynthesis with this chemical equation. We use glucose here to simplify the relationship between photosynthesis and respiration, but the direct product of photosynthesis is actually a 3-carbon sugar that can be used to make glucose. Water appears on both sides of the equation because 12 molecules are consumed and 6 molecules are newly formed during photosynthesis. We can simplify the equation by indicating only the net consumption of water. Writing the equation in this form, we can see that the overall chemical change during photosynthesis is the reverse of the one that occurs during cellular respiration, see Concept 9.1. It is important to realize that both of these metabolic processes occur in plant cells. However, as you will soon learn, chloroplasts do not synthesize sugars by simply reversing the steps of respiration. Now let's divide the photosynthetic equation by 6 to put it in its simplest possible form. Here, the brackets indicate that is not an actual sugar but represents the general formula for a carbohydrate, see Concept 5.2. In other words, we are imagining the synthesis of a sugar molecule 1 carbon at a time, with 6 repetitions theoretically adding up to a glucose molecule. Let's now see how researchers track the elements C, H, and O from the reactants of photosynthesis to the products. The splitting of water, scientific inquiry. One of the first clues to the mechanism of photosynthesis came from the discovery that the given off by plants is derived from and not from. The chloroplast splits water into hydrogen and oxygen atoms. Before this discovery, the prevailing hypothesis was that photosynthesis split carbon dioxide and then added water to the carbon. That hypothesis predicted that the release during photosynthesis came from an idea challenged in the 1930s by C. B. Van Niel, of Stanford University. Van Niel was investigating photosynthesis in bacteria that make their carbohydrate from but do not release. He concluded that, at least in these bacteria, is not split into carbon and oxygen. One group of bacteria used hydrogen sulfide rather than water for photosynthesis, forming yellow globules of sulfur as a waste product. These globules are visible in Figure 10.2e. Here is the chemical equation for photosynthesis in these sulfur bacteria. Van Niel reasoned that the bacteria split and used the hydrogen atoms to make sugar. He then generalized that idea, proposing that all photosynthetic organisms require a hydrogen source but that the source varies. Thus, Van Niel hypothesized that plants split as a source of electrons from hydrogen atoms, releasing as a byproduct. Nearly 20 years later, scientists confirmed Van Niel's hypothesis by using oxygen-18, a heavy isotope, as a tracer to follow the path of oxygen atoms during photosynthesis. The experiments showed that the produced by plants was labeled with only if water was the source of the tracer, Experiment 1. If the was introduced to the plant in the form of, the label did not turn up in the released, Experiment 2. In the following summary, green denotes labeled atoms of oxygen. A significant result of the shuffling of atoms during photosynthesis is the extraction of hydrogen from water and its incorporation into sugar. The waste product of photosynthesis is released to the atmosphere. Figure 10.4 shows the fates of all atoms in photosynthesis. Figure 10.4 tracking atoms through photosynthesis. Reactants are 6CO2 and 12H2O. Products are C6H12O6, and 6H2O, and 6O2. The atoms from are shown in magenta, and the atoms from are shown in blue. Figure 10.4 full alternative text. Photosynthesis as a redox process. Let's briefly compare photosynthesis with cellular respiration. Both processes involve redox reactions. During cellular respiration, Energy is released from sugar when electrons associated with hydrogen are transported by carriers to oxygen, forming water as a byproduct. See Concept 9.1. The electrons lose potential energy as they fall down the electron transport chain toward electronegative oxygen, and the mitochondrion harnesses that energy to synthesize ADP. See Figure 9.14. Photosynthesis reverses the direction of electron flow. Water is split, and its electrons are transferred along with hydrogen ions from the water to carbon dioxide, reducing it to sugar. Energy plus 6CO2 plus 6H2O yields C6H12O6 plus 6O2. 6,6CO2 becomes reduced as C6H12O6, and 6H2O becomes oxidized as 6O2. Because the electrons increase in potential energy as they move from water to sugar, this process requires energy. In other words, it is endergonic. This energy boost that occurs during photosynthesis is provided by light. The two stages of photosynthesis, a preview. The equation for photosynthesis is a deceptively simple summary of a very complex process. Actually, photosynthesis is not a single process, but two processes, each of which has multiple steps. These two stages of photosynthesis are known as the light reactions, the photo part of photosynthesis, and the Calvin cycle, the synthesis part. Figure 10.5. Figure 10.5. An overview of photosynthesis, cooperation of the light reactions and the Calvin cycle. An overview of photosynthesis, cooperation of the light reactions and the Calvin cycle. In the chloroplast, the thylakoid membranes, green, are the sites of the light reactions, whereas the Calvin cycle occurs in the stroma, gray. The light reactions use solar energy to make ADP and NADP, which supply chemical energy and reducing power, respectively, to the Calvin cycle. The Calvin cycle incorporates into organic molecules, which are converted to sugar. Recall that most simple sugars have formulas that are some multiple of. To visualize these processes in their cellular context, see Figure 6.32. Figure 10.5. Full alternative text. Mastering Biology. Bioflix registered animation, photosynthesis. The light reactions are the steps of photosynthesis that convert solar energy to chemical energy. Water is split, providing a source of electrons and protons, hydrogen ions, and giving off as a byproduct. Light absorbed by chlorophyll drives a transfer of the electrons and hydrogen ions from water to an acceptor called nicotinamide adenine dinucleotide phosphate, where they are temporarily stored. The electron acceptor is first cousin to which functions as an electron carrier in cellular respiration. The two molecules differ only by the presence of an extra phosphate group in the molecule. The light reactions use solar energy to reduce to NADP by adding a pair of electrons along with, and the light reactions also generate ADP, using chemiosmosis to power the addition of a phosphate group to ADP, a process called photophosphorylation. Thus, light energy is initially converted to chemical energy in the form of two compounds, NADP and ADP. NADP, a source of electrons, acts as reducing power that can be passed along to an electron acceptor, reducing it, while ADP is the versatile energy currency of cells. Notice that the light reactions produce no sugar. That happens in the second stage of photosynthesis, the Calvin cycle. The Calvin cycle is named for Melvin Calvin, who, along with his colleagues James Bassam and Andrew Benson, began to elucidate its steps in the late 1940s. The cycle begins by incorporating from the air into organic molecules already present in the chloroplast. This initial incorporation of carbon into organic compounds is known as carbon fixation. The Calvin cycle then reduces the fixed carbon to carbohydrate by the addition of electrons. The reducing power is provided by NADP, which acquired its cargo of electrons in the light reactions. To convert to carbohydrate, the Calvin cycle also requires chemical energy in the form of ADP, which is also generated by the light reactions. Thus, it is the Calvin cycle that makes sugar, but it can do so only with the help of the NADP and ADP produced by the light reactions. The metabolic steps of the Calvin cycle are sometimes referred to as the dark reactions, or light-independent reactions, because none of the steps requires light directly. Nevertheless, the Calvin cycle in most plants occurs during daylight, for only then can the light reactions provide the NADP and ADP that the Calvin cycle requires. In essence, the chloroplast uses light energy to make sugar by coordinating the two stages of photosynthesis. As Figure 10.5 indicates, the thylakoids of the chloroplast are the sites of the light reactions, while the Calvin cycle occurs in the stroma. On the outside of the thylakoids, during the light reactions, molecules of NADP pick up electrons and phosphate, respectively, and NADP and ADP are then released to the stroma, where they play crucial roles in the Calvin cycle. The two stages of photosynthesis are treated in this figure as metabolic modules that take in ingredients and crank out products. In the next two sections, we'll look more closely at how the two stages work, beginning with the light reactions. Concept check 10.2. Make connections. How do the molecules used in photosynthesis reach and enter the chloroplasts inside leaf cells? See. Concept 7.2. Explain how the use of an oxygen isotope helped elucidate the chemistry of photosynthesis. What if the Calvin cycle requires ADP and NADP, products of the light reactions? If a classmate asserted that the light reactions don't depend on the Calvin cycle and, with continual light, could just keep on producing ADP and NADP, how would you respond? For suggested answers, see Appendix A. Concept 10.3. The light reactions convert solar energy to the chemical energy of ADP and NADP. Chloroplasts are chemical factories powered by the sun. Their thylakoids transform light energy into the chemical energy of ADP and NADP. To better understand the conversion of light to chemical energy, we need to know about some important properties of light. The nature of sunlight. Light is a form of energy known as electromagnetic energy, also called electromagnetic radiation. Electromagnetic energy travels in rhythmic waves analogous to those created by dropping a pebble into a pond. Electromagnetic waves, however, are disturbances of electric and magnetic fields rather than disturbances of a material medium such as water. The distance between the crests of electromagnetic waves is called the wavelength. Wavelengths range from less than a nanometer, for gamma rays, to more than a kilometer, for radio waves. This entire range of radiation is known as the electromagnetic spectrum. Figure 10.6. The segment most important to life is the narrow band from about 380 nanometers to 740 nanometers in wavelength. This radiation is known as visible light because it can be detected as various colors by the human eye. Figure 10.6. The electromagnetic spectrum. The electromagnetic spectrum. White light is a mixture of all wavelengths of visible light. A prism can sort white light into its component colors by bending light of different wavelengths at different angles. Droplets of water in the atmosphere can act as prisms, causing a rainbow to form. Visible light drives photosynthesis. Figure 10.6. Full alternative text. The model of light as waves explains many of light's properties, but in certain respects light behaves as though it consists of discrete particles, called photons. Photons are not tangible objects, but they act like objects in that each of them has a fixed quantity of energy. The amount of energy is inversely related to the wavelength of the light. The shorter the wavelength, the greater the energy of each photon of that light. Thus, a photon of violet light packs nearly twice as much energy as a photon of red light. See Figure 10.6. Although the sun radiates the full spectrum of electromagnetic energy, the atmosphere acts like a selective window, allowing visible light to pass through while screening out a substantial fraction of other radiation. The part of the spectrum we can see visible light is also the radiation that drives photosynthesis. Photosynthetic pigments, the light receptors. When light meets matter, It may be reflected, transmitted, or absorbed. Substances that absorb visible light are known as pigments. Different pigments absorb light of different wavelengths, and the wavelengths that are absorbed disappear. If a pigment is illuminated with white light, the color we see is the color most reflected or transmitted by the pigment. If a pigment absorbs all wavelengths, it appears black. We see green when we look at a leaf because chlorophyll absorbs violet, blue and red light while transmitting and reflecting green light. Figure 10.7. The ability of a pigment to absorb various wavelengths of light can be measured with an instrument called a spectrophotometer. This machine directs beams of light of different wavelengths through a solution of the pigment and measures the fraction of the light transmitted at each wavelength. A graph plotting a pigment's light absorption versus wavelength is called an absorption spectrum. Figure 10.8. Figure 10.7. Why leaves are green, interaction of light with chloroplasts. The chlorophyll molecules of chloroplasts absorb violet, blue and red light, the colors most effective in driving photosynthesis, and reflect or transmit green light. The chlorophyll molecules of chloroplasts absorb violet, blue and red light, the colors most effective in driving photosynthesis, and reflect or transmit green light. This is why leaves appear green. Mastering biology. Animation, light energy and pigments. Figure 10.8. Research method, determining an absorption spectrum. Application. An absorption spectrum is a visual representation of how well a particular pigment absorbs different wavelengths of visible light. Absorption spectra of various chloroplast pigments help scientists decipher the role of each pigment in a plant. Technique. A spectrophotometer measures the relative amounts of light of different wavelengths absorbed and transmitted by a pigment solution. White light is separated into colors, wavelengths, by a prism. One by one, the different colors of light are passed through the sample, chlorophyll in this example. Green light and blue light are shown here. The transmitted light strikes a photoelectric tube, which converts the light energy to electricity. The electric current is measured by a galvanometer. The meter indicates the fraction of light transmitted through the sample, from which we can determine the amount of light absorbed. A diagram of a spectrometer. 10.3-4 Full alternative text. Results see. Figure 10.9 for absorption spectra of three types of chloroplast pigments. The absorption spectra of chloroplast pigments provide clues to the relative effectiveness of different wavelengths for driving photosynthesis, since light can perform work in chloroplasts only if it is absorbed. Figure 10.9 shows the absorption spectra of three types of pigments in chloroplasts. Chlorophyll A, the key light-capturing pigment that participates directly in the light reactions, the accessory pigment. Chlorophyll B, and a separate group of accessory pigments called carotenoids. The spectrum of chlorophyll A suggests that violet, blue and red light work best for photosynthesis, since they are absorbed, while green is the least effective color. This is confirmed by an action spectrum for photosynthesis. Figure 10.9b, which profiles the relative effectiveness of different wavelengths of radiation in driving the process. An action spectrum is prepared by illuminating chloroplasts with light of different colors and then plotting wavelength against some measure of photosynthetic rate, such as consumption or release. The action spectrum for photosynthesis was first demonstrated by Theodor W. Engelmann, a German botanist, in 1883. Before equipment for measuring levels had even been invented, Engelmann performed a clever experiment in which he used requiring bacteria to measure rates of photosynthesis in filamentous algae. Figure 10.9c. His results are a striking match to the modern action spectrum shown in Figure 10.9b. Figure 10.9 Inquiry, which wavelengths of light are most effective in driving photosynthesis? Experiment. Absorption and action spectra, along with a classic experiment by Theodor W. Engelmann, reveal which wavelengths of light are photosynthetically important. Results. 3 graphs of absorption and action spectra. 10.3-5 Full alternative text. Conclusion. The action spectra, confirmed by Engelmann's experiment, show which portions of the spectrum are most effective in driving photosynthesis. Data from T.W. Engelmann, Bacterium photometricum. Ein Beitrag zur vergleichenden Physiologie der Lichtwende Farbens ins Archiv. Für Physiologie 30:95, 124, 1883. Instructors, a related experimental inquiry tutorial can be assigned in Mastering Biology. Interpret the data according to the graph. Which wavelengths of light drive the highest rates of photosynthesis? Notice by comparing figures 10.9a and 10.9b that the action spectrum for photosynthesis is much broader than the absorption spectrum of chlorophyll A. The absorption spectrum of chlorophyll A alone underestimates the effectiveness of certain wavelengths in driving photosynthesis. This is partly because accessory pigments with different absorption spectra including chlorophyll B and carotenoids broaden the spectrum of colors that can be used for photosynthesis. Figure 10.10 shows the structure of chlorophyll A compared with that of chlorophyll B. A slight structural difference between them is enough to cause the two pigments to absorb at slightly different wavelengths in the red and blue parts of the spectrum, see Figure 10.9a. As a result, chlorophyll A appears blue-green and chlorophyll B olive-green under visible light. Figure 10.10 Structure of chlorophyll A and B. Structure of chlorophyll molecules in chloroplasts of plants. Figure 10.10 Full alternative text. Mastering Biology. Animation, space-filling model of chlorophyll. In the last decade, researchers have identified two other forms of chlorophyll, chlorophyll D and chlorophyll F, that absorb higher wavelengths of light. In 2018, researchers grew a species of cyanobacterium called Crococcidiopsis thermalis under only far-red light with a wavelength of 750 nanometers. They concluded that chlorophyll F was functioning in place of chlorophyll A, enabling the cyanobacterium to flourish in very shaded conditions. Higher wavelength light has less energy, so this observation extends the lower limit of energy, the red limit, needed for photosynthesis to occur. Colony of the cyanobacteria Crococcidiopsis thermalis. A micrograph captures an assortment of different colored cells. In this micrograph of cells becoming acclimated to higher wavelengths of light, cells that haven't yet acclimated and are still using chlorophyll A for photosynthesis appear purplish pink, and cells that have acclimated and are using chlorophyll F appear yellow. Other accessory pigments include carotenoids, hydrocarbons that are various shades of yellow and orange because they absorb violet and blue-green light, see Figure 10.9a. Carotenoids may broaden the spectrum of colors that can drive photosynthesis. However, a more important function of at least some carotenoids seems to be photoprotection. These compounds absorb and dissipate excessive light energy that would otherwise damage chlorophyll or interact with oxygen, forming reactive oxidative molecules that are dangerous to the cell. Interestingly, carotenoids similar to the photoprotective ones in chloroplasts have a photoprotective role in the human eye. Carrots, known for aiding night vision, are rich in carotenoids. These and related molecules are, of course, found naturally in many vegetables and fruits. They are also often advertised in health food products as phytochemicals, from the Greek phyton, plant, some of which have antioxidant properties. Plants can synthesize all the antioxidants they require, but humans and other animals must obtain some of them from their diets. Excitation of chlorophyll by light. What exactly happens when chlorophyll and other pigments absorb light? The colors corresponding to the absorbed wavelengths disappear from the spectrum of the transmitted and reflected light, but energy cannot disappear. When a molecule absorbs a photon of light, one of the molecule's electrons is elevated to an orbital where it has more potential energy, see Figure 2.6b. When the electron is in its normal orbital, the pigment molecule is said to be in its ground state. Absorption of a photon boosts an electron to an orbital of higher energy, and the pigment molecule is then said to be in an excited state. The only photons absorbed are those whose energy is exactly equal to the energy difference between the ground state and an excited state, and this energy difference varies from one kind of molecule to another. Thus, a particular compound absorbs only photons corresponding to specific wavelengths, which is why each pigment has a unique absorption spectrum. Once absorption of a photon raises an electron to an excited state, the electron cannot stay there long. Figure 10.11a. The excited state, like all high-energy states, is unstable. Generally, when isolated pigment molecules absorb light, their excited electrons drop back down to the ground state orbital in a billionth of a second, releasing their excess energy as heat. This conversion of light energy to heat is what makes the top of an automobile so hot on a sunny day. White cars are coolest because their paint reflects all wavelengths of visible light. In isolation, some pigments, including chlorophyll, emit light as well as heat after absorbing photons. As excited electrons fall back to the ground state, photons are given off, an afterglow called fluorescence. An illuminated solution of chlorophyll isolated from chloroplasts will fluoresce in the red part of the spectrum and also give off heat. Figure 10.11b. This is best seen by illuminating with ultraviolet light, which chlorophyll can also absorb, see Figures 10.6 and 10.9a. Viewed under visible light, the fluorescence would be difficult to see against the green of the solution. Figure 10.11 Excitation of isolated chlorophyll by light. Excitation of isolated chlorophyll molecule. A. Absorption of a photon causes a transition of the chlorophyll molecule from its ground state to its excited state. The photon boosts an electron to an orbital where it has more potential energy. If the illuminated molecule exists in isolation, its excited electron immediately drops back down to the ground state orbital, and its excess energy is given off as heat and fluorescence, light. B. A chlorophyll solution excited with ultraviolet light fluoresces with a red-orange glow. Figure 10.11 Full alternative text. What if a leaf containing the same concentration of chlorophyll as in the solution was exposed to the same ultraviolet light, no fluorescence would be seen. Propose an explanation for the difference in fluorescence emission between the solution and the leaf. A photosystem, a reaction center complex associated with light harvesting complexes. Chlorophyll molecules excited by the absorption of light energy produce very different results in an intact chloroplast than they do in isolation, see Figure 10.11. In their native environment of the thylakoid membrane, chlorophyll molecules are organized along with other small organic molecules and proteins into complexes called photosystems. A photosystem is composed of a reaction center complex surrounded by several light harvesting complexes. Figure 10.12. The reaction center complex is an organized association of proteins holding a special pair of chlorophyll molecules and a primary electron acceptor. Each light harvesting complex consists of various pigment molecules, which may include chlorophyll A, chlorophyll B, and multiple carotenoids, bound to proteins. The number and variety of pigment molecules enable a photosystem to harvest light over a larger surface area and a larger portion of the spectrum than could any single pigment molecule alone. Together, these light harvesting complexes act as an antenna for the reaction center complex. When a pigment molecule absorbs a photon, the energy is transferred from pigment molecule to pigment molecule within a light harvesting complex, like a human wave at a sports arena, until it is passed to the pair of chlorophyll molecules in the reaction center complex. This pair of chlorophyll molecules is special because their molecular environment, their location, and the other molecules with which they are associated enables them to use the energy from light not only to boost one of their electrons to a higher energy level, but also to transfer it to a different molecule. The primary electron acceptor, which is a molecule capable of accepting electrons and becoming reduced. Figure 10.12 The structure and function of a photosystem. The structure and function of a photosystem. Figure 10.12 Full alternative text. The solar-powered transfer of an electron from the reaction center chlorophyll pair to the primary electron acceptor is the first step of the light reactions. As soon as the chlorophyll electron is excited to a higher energy level, the primary electron acceptor captures it. This is a redox reaction. In the flask shown in Figure 10.11b, isolated chlorophyll fluoresces because there is no electron acceptor, so electrons of photo-excited chlorophyll drop right back to the ground state. In the structured environment of a chloroplast, however, an electron acceptor is readily available, and the potential energy represented by the excited electron is not dissipated as light and heat. Thus, each photosystem, a reaction center complex surrounded by light harvesting complexes, functions in the chloroplast as a unit. It converts light energy to chemical energy, which will ultimately be used for the synthesis of sugar. The thylakoid membrane is populated by two types of photosystems that cooperate in the light reactions of photosynthesis. Photosystem II, PS2, and Photosystem I, PSI. They were named in order of their discovery, but photosystem II functions first in the light reactions. Each has a characteristic reaction center complex, a particular kind of primary electron acceptor next to a special pair of chlorophyll molecules associated with specific proteins. The reaction center chlorophyll of photosystem II is known as P680 because this pigment is best at absorbing light, having a wavelength of 680 nanometers, in the red part of the spectrum. The chlorophyll at the reaction center complex of photosystem I is called P700 because it most effectively absorbs light of wavelength 700 nanometers, in the far red part of the spectrum. These two pigments, P680 and P700, are nearly identical chlorophyll molecules. However, their association with different proteins in the thylakoid membrane affects the electron distribution in the two pigments and accounts for the slight differences in their light-absorbing properties. Now let's see how the two types of photosystems work together in using light energy to generate ADP and NADP, the two main products of the light reactions. Linear electron flow. Light drives the synthesis of ADP and NADP by energizing the two types of photosystems embedded in the thylakoid membranes of chloroplasts. The key to this energy transformation is a flow of electrons through the photosystems and other molecular components built into the thylakoid membrane. This is called linear electron flow, and it occurs during the light reactions of photosynthesis, as shown in Figure 10.13. The numbered steps in the text correspond to the numbered steps in the figure. Figure 10.13 How linear electron flow during the light reactions generates ADP and NADP. How linear electron flow during the light reactions generates ATP and NADPH. The gold arrows trace the flow of light-driven electrons from water to NADP. The black arrows trace the transfer of energy from pigment molecule to pigment molecule. To see these proteins in their cellular context, see Figure 6.32b. Figure 10.13 Full alternative text. Mastering biology. Figure walkthrough. A photon of light strikes one of the pigment molecules in a light harvesting complex of PS2, boosting one of its electrons to a higher energy level. As this electron falls back to its ground state, an electron in a nearby pigment molecule is simultaneously raised to an excited state. The process continues, with the energy being relayed to other pigment molecules until it reaches the P680 pair of chlorophyll molecules in the PS2 reaction center complex. It excites an electron in this pair of chlorophylls to a higher energy state. This electron is transferred from the excited P680 to the primary electron acceptor. We can refer to the resulting form of P680, missing the negative charge of an electron, as an enzyme catalyzes the splitting of a water molecule into two electrons, two hydrogen ions, and an oxygen atom. The electrons are supplied one by one to the pair, each electron replacing one transferred to the primary electron acceptor. Is the strongest biological oxidizing agent known, its electron hole must be filled. This greatly facilitates the transfer of electrons from the split water molecule. They are released into the thylakoid space, interior of the thylakoid. The oxygen atom immediately combines with an oxygen atom generated by the splitting of another water molecule, forming. Each photoexcited electron passes from the primary electron acceptor of PS2 to PSI via an electron transport chain, the components of which are similar to those of the electron transport chain that functions in cellular respiration. The electron transport chain between PS2 and PSI is made up of the electron carrier plastoquinone, PQ, a cytochrome complex, and a protein called plastocyanin, PC. Each component carries out redox reactions as electrons flow down the electron transport chain, releasing free energy that is used to pump protons into the thylakoid space, contributing to a proton gradient across the thylakoid membrane. The potential energy stored in the proton gradient is used to make ADP in a process called chemiosmosis, to be discussed shortly. Meanwhile, light energy has been transferred via light harvesting complex pigments to the PSI reaction center complex, exciting an electron of the P700 pair of chlorophyll molecules located there. The photoexcited electron is then transferred to PSI's primary electron acceptor, creating an electron hole in the P700 which we now can call. In other words, can now act as an electron acceptor, accepting an electron that reaches the bottom of the electron transport chain from PS2. Photoexcited electrons are passed in a series of redox reactions from the primary electron acceptor of PSI down a second electron transport chain through the protein ferredoxin, FD. This chain does not create a proton gradient and thus does not produce ADP. The enzyme reductase catalyzes the transfer of electrons from FD to 2 electrons are required for its reduction to NADP. Electrons in NADP are at a higher energy level than they are in water, where they started, so they are more readily available for the reactions of the Calvin cycle. This process also removes N from the stroma. The energy changes of electrons during their linear flow through the light reactions are shown in a mechanical analogy in Figure 10.14. Although the scheme shown in Figures 10.13 and 10.14 may seem complicated, do not lose track of the big picture. The light reactions use solar power to generate ADP and NADP, which provide chemical energy and reducing power, respectively, to the carbohydrate synthesizing reactions of the Calvin cycle. Figure 10.14, a mechanical analogy for linear electron flow during the light reactions. A mechanical analogy for linear electron flow during the light reactions. Figure 10.14, full alternative text. Cyclic electron flow. In certain cases, photoexcited electrons can take an alternative path called cyclic electron flow, which uses photosystem I but not photosystem II. You can see in Figure 10.15 that cyclic flow is a short circuit. The electrons cycle back from ferredoxin, Fd, to the cytochrome complex, then via a plastocyanin molecule, Pc, to AP700 chlorophyll in the PSI reaction center complex. There is no production of NADP and no release of oxygen that results from this process. On the other hand, cyclic flow does generate ADP. Figure 10.15 cyclic electron flow. Cyclic electron flow. Photoexcited electrons from PSI are occasionally shunted back from ferredoxin, Fd, to chlorophyll via the cytochrome complex and plastocyanin, Pc. This electron shunt supplements the supply of ADP, via chemiosmosis, but produces no NADP. The shadow of linear electron flow is included in the diagram for comparison with the cyclic route. The two Fd molecules in this diagram are actually one and the same, the final electron carrier in the electron transport chain of PSI, although it is depicted twice to clearly show its role in two parts of the process. Figure 10.15 full alternative text. Visual skills look at Figure 10.14 and explain how you would alter it to show a mechanical analogy for cyclic electron flow. Rather than having both PS2 and PSI, several of the currently existing groups of photosynthetic bacteria are known to have a single photosystem related to either PS2 or PSI. For these species, which include the purple sulfur bacteria, see Figure 10.2e, and the green sulfur bacteria, cyclic electron flow is the one and only means of generating ADP during the process of photosynthesis. Evolutionary biologists hypothesize that these bacterial groups are descendants of ancestral bacteria in which photosynthesis first evolved, in a form similar to cyclic electron flow. Cyclic electron flow can also occur in photosynthetic species that possess both photosystems. This includes some prokaryotes, such as the cyanobacteria shown in Figure 10.2d, as well as the eukaryotic photosynthetic species that have been tested thus far. Although the process is probably in part an evolutionary leftover, research suggests it plays at least one beneficial role for these organisms. Plants with mutations that render them unable to carry out cyclic electron flow are capable of growing well in low light, but do not grow well where light is intense. This is evidence for the idea that cyclic electron flow may be photoprotective. Later you'll learn more about cyclic electron flow as it relates to a particular adaptation of photosynthesis, plants, see Concept 10.5. Whether ADP synthesis is driven by linear or cyclic electron flow, the actual mechanism is the same. Before we move on to consider the Calvin cycle, let's review chemiosmosis, the process that uses membranes to couple redox reactions to ADP production. A comparison of chemiosmosis in chloroplasts and mitochondria. Chloroplasts and mitochondria generate ADP by the same basic mechanism, chemiosmosis, see Figure 9.14. An electron transport chain pumps protons across a membrane as electrons are passed through a series of carriers that have progressively more affinity for electrons. Thus, electron transport chains transform redox energy to a proton motive force, potential energy stored in the form of an gradient across a membrane. An ADP synthase complex in the same membrane couples the diffusion of hydrogen ions down their gradient to the phosphorylation of ADP, forming ADP. Some of the electron carriers, including the iron-containing proteins called cytochromes, are very similar in mitochondria and chloroplasts, see Figure 6.32b and c. The ADP synthase complexes of the two organelles are also quite similar. But there are noteworthy differences between photophosphorylation in chloroplasts and oxidative phosphorylation in mitochondria. Both work by way of chemiosmosis, but in chloroplasts, the high-energy electrons drop down the transport chain come from water, while in mitochondria, they are extracted from organic molecules, which are thus oxidized. Chloroplasts do not need molecules from food to make ADP, Their photosystems capture light energy and use it to drive the electrons from water to the top of the transport chain. In other words, mitochondria use chemiosmosis to transfer chemical energy from food molecules to ADP, whereas chloroplasts use it to transform light energy into chemical energy in ADP. Although the spatial organization of chemiosmosis differs slightly between chloroplasts and mitochondria, it is easy to see similarities in the two. Figure 10.16. Electron transport chain proteins in the inner membrane of the mitochondrion pump protons from the mitochondrial matrix out to the intermembrane space, which then serves as a reservoir of hydrogen ions. Similarly, electron transport chain proteins in the thylakoid membrane of the chloroplast pump protons from the stroma into the thylakoid space, which functions as the reservoir. If you imagine the cristae of mitochondria pinching off from the inner membrane, this may help you see how the thylakoid space and the intermembrane space are comparable spaces in the two organelles, while the mitochondrial matrix is analogous to the stroma of the chloroplast. Figure 10.16 Comparison of chemiosmosis in mitochondria and chloroplasts. Comparison of chemiosmosis in mitochondria and chloroplasts. In both kinds of organelles, electron transport chains pump protons across a membrane from a region of low concentration, light gray in this diagram, to one of high concentration, dark gray. The protons then diffuse back across the membrane through ADP synthase, driving the synthesis of ADP. Figure 10.16 Full alternative text. Make connections. Describe how you would change the pH in order to artificially cause ADP synthesis. A. Outside an isolated mitochondrion, assume can freely cross the outer membrane. C. Figure 9.14, and, B, in the stroma of a chloroplast. Explain. In the mitochondrion, protons diffuse down their concentration gradient from the intermembrane space through ADP synthase to the matrix, driving ADP synthesis. In the chloroplast, ADP is synthesized as the hydrogen ions diffuse from the thylakoid space back to the stroma through ADP synthase complexes. Figure 10.17, whose catalytic knobs are on the stroma side of the membrane. Thus, ADP forms in the stroma, where it is used to help drive sugar synthesis during the Calvin cycle. Figure 10.17 The light reactions, organization of the thylakoid membrane. The light reactions and chemiosmosis, the organization of the thylakoid membrane. Figure 10.17 Full alternative text. Visual skills. Which three steps in the figure contribute to the gradient across the thylakoid membrane? Mastering biology. Animation, the light reactions. The proton gradient, or pH gradient, across the thylakoid membrane is substantial. When chloroplasts in an experimental setting are illuminated, the pH in the thylakoid space drops to about 5, the concentration increases, and the pH in the stroma increases to about 8, the concentration decreases. This gradient of 3 pH units corresponds to a 1,000-fold difference in concentration. If the lights are then turned off, the pH gradient is abolished, but it can quickly be restored by turning the lights back on. Experiments such as this provided strong evidence in support of the chemiosmotic model. The currently accepted model for the organization of the light reaction machinery within the thylakoid membrane is based on several research studies. Each of the molecules and molecular complexes in Figure 10.17 is present in numerous copies in each thylakoid. Notice that NADP, like ADP, is produced on the side of the membrane facing the stroma, where the Calvin cycle reactions take place. Let's step back and see the big picture by summarizing the light reactions. Electron flow pushes electrons from water, where they are at a state of low potential energy, ultimately to NADP, where they are stored at a state of high potential energy. The light-driven electron flow also generates ADP. Thus, the equipment of the thylakoid membrane converts light energy to chemical energy stored in ADP, and NADP is produced as a byproduct. Let's now see how the enzymes of the Calvin cycle use the products of the light reactions to synthesize sugar from Mastering biology. Bioflix registered animation, the light reactions. Concept check 10.3. What color of light is least effective in driving photosynthesis? Explain. In the light reactions, what is the initial electron donor? Where do the electrons finally end up? What if, in an experiment, isolated chloroplasts placed in an illuminated solution with the appropriate chemicals can carry out ADP synthesis? Predict what would happen to the rate of synthesis if a compound is added to the solution that makes membranes freely permeable to hydrogen ions. For suggested answers, see Appendix A. Concept 10.4 The Calvin cycle uses the chemical energy of ADP and NADP to reduce. The equation shows co-subscript 2 to sugar. The Calvin cycle takes place in the stroma. It is similar to the citric acid cycle in that a starting material is regenerated after some molecules enter and others exit the cycle. However, the citric acid cycle is catabolic, oxidizing acetyl-CoA and using the energy to synthesize ADP, see Figure 9.11, while the Calvin cycle is anabolic, building carbohydrates from smaller molecules and consuming energy. Carbon enters the Calvin cycle in the form of CO2 and leaves in the form of sugar. The cycle spends ADP as an energy source and consumes NADP as reducing power for adding high-energy electrons to make the sugar. As we mentioned in Concept 10.2, the carbohydrate produced directly from the Calvin cycle is not glucose. It is actually a 3-carbon sugar called glyceraldehyde 3-phosphate, G3P. For the net synthesis of one molecule of G3P, the cycle must take place 3 times, fixing 3 molecules of 1 per turn of the cycle. Recall that the term carbon fixation refers to the initial incorporation of into organic material. As we trace the steps of the Calvin cycle, keep in mind that we are following 3 molecules of through the reactions. Figure 10.18 divides the Calvin cycle into 3 phases: carbon fixation, reduction, and regeneration of the acceptor. Figure 10.18 the Calvin cycle. The Calvin cycle. This diagram summarizes 3 turns of the cycle, tracking carbon atoms, shown as dark gray spheres. The 3 phases of the cycle correspond to the phases discussed in the text. For every 3 molecules of that enter the cycle, the net output is 1 molecule of glyceraldehyde 3-phosphate, G3P, a 3-carbon sugar. The light reactions sustain the Calvin cycle by regenerating the required ADP and NADP. Figure 10.18 full alternative text. Mastering Biology. Animation, the Calvin cycle. Bioflix registered animation, the Calvin cycle. Phase 1, carbon fixation. The Calvin cycle incorporates each molecule, one at a time, by attaching it to a 5-carbon sugar named ribulose bisphosphate, which is abbreviated RUBP. The enzyme that catalyzes this first step is RUBP carboxylase oxygenase, or Rubisco, C. Figure 6.32c. This is the most abundant protein in chloroplasts and is also thought to be the most abundant protein on Earth. The product of the reaction is a 6-carbon intermediate that is short-lived because it is so energetically unstable that it immediately splits in half, forming 2 molecules of 3-phosphoglycerate, 4 each fixed. Phase 2, reduction. Each molecule of 3-phosphoglycerate receives an additional phosphate group from ADP, becoming 1,3-bisphosphoglycerate. Next, a pair of electrons donated from NADP produces 1,3-bisphosphoglycerate, which also loses a phosphate group in the process, becoming glyceraldehyde 3-phosphate, G3P. Specifically, the electrons from NADP produce a carboxyl group on 1,3-bisphosphoglycerate to the aldehyde group of G3P, which stores more potential energy. G3P is a sugar, the same 3-carbon sugar formed in glycolysis by the splitting of glucose, C. Figure 9.8. Notice in Figure 10.18 that for every 3 molecules of that enter the cycle, there are 6 molecules of G3P formed. But only 1 molecule of this 3-carbon sugar can be counted as a net gain of carbohydrate because the rest are required to complete the cycle. The cycle began with 15 carbons worth of carbohydrate in the form of 3 molecules of the 5-carbon sugar RUBP. Now there are 18 carbons worth of carbohydrate in the form of 6 molecules of G3P. 1 molecule exits the cycle to be used by the plant cell, but the other 5 molecules must be recycled to regenerate the 3 molecules of RUBP. Phase 3, regeneration of the acceptor, RUBP. In a complex series of reactions, the carbon skeletons of 5 molecules of G3P are rearranged by the last steps of the Calvin cycle into 3 molecules of RUBP. To accomplish this, the cycle spends 3 more molecules of ADP. The RUBP is now prepared to receive again, and the cycle continues. For the net synthesis of one G3P molecule, the Calvin cycle consumes a total of 9 molecules of ADP and 6 molecules of NADP. The light reactions regenerate the ADP and NADP. The G3P spun off from the Calvin cycle becomes the starting material for metabolic pathways that synthesize other organic compounds, including glucose, from 2 molecules of G3P, sucrose, and other carbohydrates. Neither the light reactions nor the Calvin cycle alone can make sugar from. Photosynthesis is an emergent property of the intact chloroplast, which integrates the 2 stages of photosynthesis. Concept check 10.4. To synthesize one glucose molecule, the Calvin cycle uses molecules of ADP, and molecules of NADP. How are the large numbers of ADP and NADP molecules used during the Calvin cycle consistent with the high value of glucose as an energy source? What if? Consider a poison that inhibits an enzyme of the Calvin cycle. Do you think such a poison will also inhibit the light reactions? Explain. Draw a simple version of the Calvin cycle that shows only the number of carbon atoms at each step, using numerals rather than gray spheres, for example, at the beginning of the cycle. Explain how the total number of carbon atoms remains constant for every 3 turns of the cycle, noting the forms in which the carbon atoms enter and leave the cycle. Make connections review. Figures 9.8 and 10.18, and then discuss the roles of intermediate and product played by glyceraldehyde 3-phosphate, G3P, in the two processes shown in these figures. For suggested answers, see Appendix A. Concept 10.5 Alternative mechanisms of carbon fixation have evolved in hot, arid climates. Evolution. Ever since plants first moved onto land about 475 million years ago, they have been adapting to the problems of terrestrial life, particularly the problem of dehydration. In Concept 36.4, we will consider anatomical adaptations that help plants conserve water, while in this chapter we are concerned with metabolic adaptations. The solutions often involve trade-offs. An important example is the balance between photosynthesis and the prevention of excessive water loss from the plant. The CO2 required for photosynthesis enters a leaf, and the resulting O2 exits, via stomata, the pores on the leaf surface, see Figure 10.3. However, stomata are also the main avenues of transpiration, the evaporative loss of water from leaves. On a hot, dry day, most plants close their stomata, a response that conserves water but also reduces CO2 levels, with stomata even partially closed. CO2 concentrations begin to decrease in the air spaces within the leaf, and the concentration of O2 released from the light reactions begins to increase. These conditions within the leaf favor an apparently wasteful process called photorespiration. Photorespiration, an evolutionary relic. In most plants, initial fixation of carbon occurs via rubisco, the Calvin cycle enzyme that adds to ribulose bisphosphate. Such plants are called plants because the first organic product of carbon fixation is a 3-carbon compound, 3-phosphoglycerate, see Figure 10.18. Plants include important agricultural plants such as rice, wheat, and soybeans. When their stomata close on hot, dry days, plants produce less sugar because the declining level of in the leaf starves the Calvin cycle. In addition, rubisco is capable of binding in place of. As becomes scarce within the air spaces of the leaf and builds up, rubisco adds to the Calvin cycle instead of. The product splits, and a 2-carbon compound leaves the chloroplast. Peroxisomes and mitochondria within the plant cell rearrange and split this compound, releasing. The process is called photorespiration because it occurs in the light, photo, and consumes while producing, respiration. However, unlike normal cellular respiration, photorespiration uses ATP rather than generating it. And unlike photosynthesis, photorespiration produces no sugar. In fact, photorespiration decreases photosynthetic output by siphoning organic material from the Calvin cycle and releasing that would otherwise be fixed. This can eventually be fixed if it is still in the leaf once the concentration builds up to a high enough level. In the meantime, though, the process is energetically costly, much like a hamster running on its wheel. How can we explain the existence of a metabolic process that seems to be counterproductive for the plant? According to one hypothesis, photorespiration is evolutionary baggage, a metabolic relic from a much earlier time when the atmosphere had less and more than it does today. In the ancient atmosphere that prevailed when rubisco first evolved, the ability of the enzyme's active site to bind would have made little difference. The hypothesis suggests that modern rubisco retains some of its chance affinity for, which is now so concentrated in the atmosphere that a certain amount of photorespiration is inevitable. There is also some evidence that photorespiration may provide protection against the damaging products of the light reactions, which build up when the Calvin cycle slows due to low. In many types of plants, including a significant number of crop plants, photorespiration drains away as much as 50% of the carbon fixed by the Calvin cycle. Indeed, if photorespiration could be reduced in certain plant species without otherwise affecting photosynthetic productivity, crop yields and food supplies might increase. In some plant species, alternate modes of carbon fixation have evolved that minimize photorespiration and optimize the Calvin cycle even in hot, arid climates. The two most important of these photosynthetic adaptations are photosynthesis and crassulation acid metabolism, CAM. Plants. The Plants are so named because they preface the Calvin cycle with an alternate mode of carbon fixation that forms a 4-carbon compound as its first product. The pathway is believed to have evolved independently at least 45 separate times and is used by several thousand species in at least 19 plant families. Among the plants important to agriculture are sugarcane and corn, members of the grass family. When the weather is hot and dry, a plant partially closes its stomata, conserving water but reducing the concentration in the leaves. However, sugar is still made because plants use a multi-step process that operates even under low conditions. Photosynthesis begins in mesophyll cells but is completed in bundle sheath cells, cells that are arranged into tightly packed sheaths around the veins of the leaf. Figure 10.19. In leaves, the loosely arranged mesophyll cells are located between the bundle sheath cells and the leaf surface, no more than 2 to 3 cells away from the bundle sheath cells. In mesophyll cells, is incorporated into organic compounds that then move into the bundle sheath cells, where the Calvin cycle takes place. See the numbered steps in Figure 10.19, which are also described here. Figure 10.19 Leaf anatomy and the pathway. C4 Leaf anatomy and the C4 pathway. The structure and biochemical functions of the leaves of plants are an evolutionary adaptation to hot, dry climates. This adaptation maintains a concentration in the bundle sheath cells that favors photosynthesis over photorespiration. Figure 10.19 Full alternative text. The first step is carried out by an enzyme present only in mesophyll cells called PEP carboxylase, which has a much higher affinity for than does rubisco, and no affinity for. This enzyme adds to phosphonol pyruvate, PEP, forming the 4-carbon product oxalostate. It does this even under conditions of lower concentration and relatively higher concentration. After the is fixed in the mesophyll cells, the 4-carbon products, malate in the example shown in Figure 10.19, are exported to bundle sheath cells through plasmodesmata, see Figure 6.29. Within the bundle sheath cells, an enzyme releases from the 4-carbon compounds, the is refixed into organic material by rubisco and the Calvin cycle. The same reaction regenerates pyruvate, which is transported to mesophyll cells. There, ATP is used to convert pyruvate to PEP, which can then accept addition of another, allowing the reaction cycle to continue. This ATP can be thought of, in a sense, as the price of concentrating in the bundle sheath cells. To generate this extra ATP, bundle sheath cells carry out cyclic electron flow, the process described earlier in this chapter, see Figure 10.15. In fact, these cells contain PSI but no PS2, so cyclic electron flow is their only photosynthetic mode of generating ATP. In effect, the mesophyll cells of a plant pump into the bundle sheath cells, keeping the concentration in those cells high enough for rubisco to bind rather than. The cyclic series of reactions involving PEP carboxylase and the regeneration of PEP can be thought of as an ATP-powered pump that concentrates where it can be fixed. In this way, photosynthesis spends ATP energy to minimize photorespiration and enhance sugar production. This adaptation is especially advantageous in hot regions with intense sunlight, and it is in such environments that plants evolved and thrive today. The concentration of in the atmosphere has drastically increased since the Industrial Revolution began in the 1800s, and it continues to rise today due to human activities such as the burning of fossil fuels, see Concept 1.1. The resulting global climate change, including an increase in average temperatures around the planet, may have far-reaching effects on plant species. Scientists are concerned that increasing concentration and temperature may affect plants differently, thus changing the relative abundance of these species in a given plant community. Which type of plant would stand to gain more from increasing levels? Recall that in plants, the binding of rather than by rubisco leads to photorespiration, lowering the efficiency of photosynthesis. Plants overcome this problem by concentrating in the bundle sheath cells at the cost of ATP. Rising levels should benefit plants by lowering the amount of photorespiration that occurs. At the same time, rising temperatures have the opposite effect, increasing photorespiration. Other factors such as water availability may also come into play. In contrast, many plants could be largely unaffected by increasing levels or temperature. Researchers have investigated aspects of this question in several studies. You can work with data from one such experiment in the scientific skills exercise. In different regions, the particular combination of concentration and temperature is likely to alter the balance of plants in varying ways. The effects of such a widespread and variable change in community structure are unpredictable and thus a cause of legitimate concern. Scientific skills exercise. Making scatter plots with regression lines. Does atmospheric concentration affect the productivity of agricultural crops? The atmospheric concentration of has been rising globally, and scientists wondered whether this would affect and plants differently. In this exercise, you will make a scatter plot to examine the relationship between concentration and growth of both corn, maize, a crop plant, and velvet leaf, a weed found in cornfields. Corn plant with velvet leaf around it. Corn plant surrounded by invasive velvet leaf plants. How the experiment was done. For 45 days, researchers grew corn and velvet leaf plants under controlled conditions, where all plants received the same amounts of water and light. The plants were divided into 3 groups, and each was exposed to a different concentration of in the air, 350, 600, or 1000 ppm, parts per million. Data from the experiment. The table shows the dry mass, in grams, of corn and velvet leaf plants grown at the 3 concentrations of. The dry mass values are averages calculated from the leaves, stems, and roots of 8 plants. 350 ppm, 600 ppm, 1000 ppm. Average dry mass of one corn plant, G. 91, 89, 80. Average dry mass of one velvet leaf plant, G. 35, 48, 54. Data from D.T. Patterson and E.P. Flint, Potential Effects of Global Atmospheric Enrichment on the Growth and Competitiveness of N-Wheat and Crop Plants, Weed Science 28, 171, 75, 1980. Interpret the data. To explore the relationship between the 2 variables, it is useful to graph the data in a scatter plot, and then draw a regression line. A. First, place labels for the dependent and independent variables on the appropriate axis. Explain your choices. B. Plot the data points for corn and velvet leaf using different symbols for each set of data, and add a key for the 2 symbols. For additional information about graphs, see the Scientific Skills Review in Appendix D. Draw a best fit line for each set of points. A best fit line does not necessarily pass through all or even most points. Instead, it is a straight line that passes as close as possible to all data points from that set. Draw a best fit line for each set of data. Because placement of the line is a matter of judgment, 2 individuals may draw 2 slightly different lines for a given set of points. The line that actually fits best, a regression line, can be identified by squaring the distances of all points to any candidate line, then selecting the line that minimizes the sum of the squares. See the graph in the Scientific Skills exercise in Chapter 3 for an example of a linear regression line. Excel or other software programs, including those on a graphing calculator, can plot a regression line once data points are entered. Using either Excel or a graphing calculator, enter the data points for each data set and have the program draw the 2 regression lines. Compare them to the lines you drew. Describe the trends shown by the regression lines in your scatter plot. A. Compare the relationship between increasing concentration of and the dry mass of corn to that for velvet leaf. B. Considering that velvet leaf is a weed invasive to cornfields, predict how increased concentration may affect interactions between the 2 species. Based on the data in the scatter plot, estimate the percentage change in dry mass of corn and velvet leaf plants if atmospheric concentration increased from 390 ppm, current levels, to 800 ppm. A. What is the estimated dry mass of corn and velvet leaf plants at 390 ppm? 800 ppm. B. To calculate the percentage change in mass for each plant, subtract the mass at 390 ppm from the mass at 800 ppm, change in mass, divide by the mass at 390 ppm, initial mass, and multiply by 100. What is the estimated percentage change in dry mass for corn? For velvet leaf? C. Do these results support the conclusion from other experiments that plants grow better than plants under increased concentration? Why or why not? Instructors, a version of the Scientific Skills exercise can be assigned in Mastering Biology. Photosynthesis is considered more efficient than photosynthesis because it uses less water and resources. On our planet today, the world population and demand for food are rapidly increasing. At the same time, the amount of land suitable for growing crops is decreasing due to the effects of global climate change, which include an increase in sea level as well as a hotter, drier climate in many regions. To address issues of food supply, scientists in the Philippines have been working on genetically modifying rice, an important food staple that is a crop, so that it can instead carry out photosynthesis. Results so far seem promising, and these researchers estimate that the yield of rice might be 30 to 50% higher than rice with the same input of water and resources. Chem plants. A second photosynthetic adaptation to arid conditions has evolved in pineapples, many cacti, and other succulent, water-storing plants, such as aloe and jade plants. These plants open their stomata at night and close them during the day, just the reverse of how other plants behave. Closing stomata during the day helps desert plants conserve water, but it also prevents from entering the leaves. During the night, when their stomata are open, these plants take up and incorporate it into a variety of organic acids. This mode of carbon fixation is called crassulation acid metabolism, or CAM, after the plant family Crassulaceae, the succulents in which the process was first discovered. The mesophyll cells of CAM plants store the organic acids they make during the night in their vacuoles until morning, when the stomata close. During the day, when the light reactions can supply ATP and NAD for the Calvin cycle, is released from the organic acids made the night before to become incorporated into sugar in the chloroplasts. Notice in Figure 10.20 that the CAM pathway is similar to the pathway in that is first incorporated into organic intermediates before it enters the Calvin cycle. The difference is that in plants, the initial steps of carbon fixation are separated structurally from the Calvin cycle, whereas in CAM plants, the two steps occur within the same cell but at separate times. Keep in mind that CAM and plants all eventually use the Calvin cycle to make sugar from carbon dioxide. Mastering biology. Animation, photosynthesis in dry climates. Figure 10.20 and CAM photosynthesis compared. C4 and CAM photosynthesis compared. In C4 plants there is a spatial separation of steps and in CAM plants there is a temporal separation of steps. The NCAM pathways are two evolutionary solutions to the problem of maintaining photosynthesis with stomata partially or completely closed on hot, dry days. Both adaptations are characterized by 1. Preliminary incorporation of into organic acids, followed by 2. Transfer of to the Calvin cycle. Figure 10.20 full alternative text. Concept check 10.5. Describe how photorespiration lowers photosynthetic output for plants. The presence of only PSI, not PS2, in the bundle sheath cells of plants has an effect on concentration. What is that effect, and how might that benefit the plant? Make connections refer to the discussion of ocean acidification in Concept 3.3. Ocean acidification and changes in the distribution of N plants may seem to be two very different problems, but what do they have in common? Explain. What if? How would you expect the relative abundance of versus NCAM species to change in a geographic region whose climate becomes much hotter and drier, with no change in concentration? For suggested answers, see Appendix A. Concept 10.6 Photosynthesis is essential for life on Earth, a review. Mastering biology. Bioflix registered animation, photosynthesis. In this chapter, we have followed photosynthesis from photons to food. The light reactions capture solar energy and use it to make ATP and transfer electrons from H2O to NADP+, forming NADP. The Calvin cycle uses the ATP and NADP to produce sugar from CO2. The energy that enters the chloroplasts as sunlight becomes stored as chemical energy in organic compounds. The entire process is reviewed visually in Figure 10.21, where photosynthesis is also put in its natural context. Figure 10.21 A review of photosynthesis. A review of photosynthesis using the cells of a tree as an example. This diagram shows the main reactants and products of photosynthesis as they move through the tissues of a tree, left, and a chloroplast, right. Figure 10.21 Full alternative text. Mastering biology. HHMI animation, photosynthesis. MP3 tutor, photosynthesis. As for the fates of photosynthetic products, enzymes in the chloroplast and cytosol convert the G3P made in the Calvin cycle to many other organic compounds. In fact, the sugar made in the chloroplast supplies the entire plant with chemical energy and carbon skeletons for the synthesis of all the major organic molecules of plant cells. About 50% of the organic material made by photosynthesis is consumed as fuel for cellular respiration in plant cell mitochondria. Technically, green cells are the only autotrophic parts of the plant. The rest of the plant depends on organic molecules exported from leaves through veins, see. Figure 10.21, top. In most plants, carbohydrate is transported out of the leaves to the rest of the plant in the form of sucrose, a disaccharide. After arriving at non-photosynthetic cells, the sucrose provides raw material for cellular respiration and a multitude of anabolic pathways that synthesize proteins, lipids, and other products. A considerable amount of sugar in the form of glucose is linked together to make the polysaccharide cellulose, see. Figure 5.6c, especially in plant cells that are still growing and maturing. Cellulose, the main ingredient of cell walls, is the most abundant organic molecule in the plant and probably on the surface of the planet. Most plants and other photosynthesizers make more organic material per day than they need to use as respiratory fuel and precursors for biosynthesis. For example, flowering plants stockpile the extra sugar in the form of starch, storing some in the chloroplasts and some in cells of roots, tubers, seeds, and fruits. In accounting for the use of food molecules produced by photosynthesis, note that most flowering plants lose leaves, roots, stems, fruits, and often their entire bodies to heterotrophs, including humans. On a global scale, photosynthesis is the process responsible for the presence of in our atmosphere. Furthermore, although each chloroplast is minuscule, their collective productivity in terms of food production is enormous. Photosynthesis makes an estimated 150 billion metric tons of carbohydrate per year. A metric ton is 1,000 kilograms, about 1.1 tons. That's organic matter equivalent in mass to a stack of about 60 trillion biology textbooks, and the stack would reach 17 times the distance from Earth to the Sun. No chemical process is more important than photosynthesis to the welfare of life on Earth. In chapters 5 through 10, you have learned about many activities of cells. Figure 10.22 integrates these cellular processes into the context of a working plant cell. As you study the figure, reflect on how each process fits into the big picture. As the most basic unit of living organisms, a cell performs all functions characteristic of life. Figure 10.22 Make connections, the working cell. The figure illustrates how a generalized plant cell functions, integrating the cellular activities discussed in chapters 5 to 10. Figure 10.22 Full alternative text. Make connections. The first enzyme that functions in glycolysis is hexokinase, see. Figure 6.32a. How is hexokinase produced in this plant cell? Specify the locations of each step of the process. Where does hexokinase function? See. Figures 5.18, 5.22, and 9.8. Mastering biology. Bioflix registered animation, tour of an animal cell. Bioflix registered animation, tour of a plant cell. Concept check 10.6. Make connections. How do plants use the sugar they produce during photosynthesis to directly power the work of the cell? Provide some examples of cellular work. See. Figures 8.10, 8.11, and 9.5. For suggested answers, see Appendix A. Summary of key concepts. Go to Mastering Biology for assignments, the e-text, the study area, and dynamic study modules. To review key terms, go to the vocabulary self-quiz in the Mastering Biology e-text or study area, or go to goo.gl/zkjz9t. Concept 10.1 Photosynthesis feeds the biosphere, pp. 188. Photosynthesis converts the energy of sunlight into chemical energy stored in sugars. Autotrophs, producers, produce their organic molecules from and other inorganic raw materials. Plants are photoautotrophs that use light energy to do this. Heterotrophs, consumers, including decomposers, are unable to make their own food and live on compounds produced by others. Using the terms producers, consumers, and decomposers, explain how photosynthesis feeds organisms, either directly or indirectly. Concept 10.2 Photosynthesis converts light energy to the chemical energy of food, pp. 189. 192. In plants and other eukaryotic autotrophs, photosynthesis occurs in chloroplasts, organelles containing thylakoids. Stacks of thylakoids form grana. Photosynthesis is summarized as: chloroplasts split water into hydrogen and oxygen, incorporating the electrons of hydrogen into sugar molecules. Photosynthesis is a redox process, is oxidized, and is reduced. The light reactions in the thylakoid membrane split water, releasing, producing ADP, and forming NADP. The Calvin cycle in the stroma forms sugar from, using ADP for energy and NADP for reducing power. Compare the roles of N in cellular respiration and photosynthesis. Concept 10.3 The light reactions convert solar energy to the chemical energy of ADP and NADP, pp. 192. 201. Light is a form of electromagnetic energy. The colors we see as visible light include those wavelengths that drive photosynthesis. A pigment absorbs light of specific wavelengths. Chlorophyll is the main photosynthetic pigment in plants. Other accessory pigments absorb different wavelengths of light and pass the energy on to chlorophyll A. A pigment goes from a ground state to an excited state when a photon of light boosts one of the pigment's electrons to a higher energy orbital. This excited state is unstable. Electrons from isolated pigments tend to fall back to the ground state, giving off heat and/or light. A photosystem is composed of a reaction center complex surrounded by light harvesting complexes that funnel the energy of photons to the reaction center complex. When a special pair of reaction center chlorophyll molecules absorbs energy, one of its electrons is boosted to a higher energy level and transferred to the primary electron acceptor. Photosystem II contains P680 chlorophyll molecules in the reaction center complex. Photosystem I contains P700 molecules. Linear electron flow during the light reactions uses both photosystems and produces NADP, ADP, and oxygen. How linear electron flow during the light reactions generates ATP and NADPH. 10.2 to 10 Full alternative text. Cyclic electron flow employs only one photosystem, producing ADP but no NADP or. During chemiosmosis in both mitochondria and chloroplasts, electron transport chains generate an gradient across a membrane. ADP synthase uses this proton motive force to make ADP. The absorption spectrum of chlorophyll differs from the action spectrum of photosynthesis. Explain this observation. Concept 10.4 The Calvin cycle uses the chemical energy of ADP and NADP to reduce to sugar, pp. 201. 202. The Calvin cycle occurs in the stroma, using electrons from NADP and energy from ADP. One molecule of G3P exits the cycle per 3 molecules fixed and is converted to glucose and other organic molecules. Sketch of the Calvin cycle showing the changes in carbon molecules. 10.2 to 11 Full alternative text. Draw it on the diagram above. Draw where ADP and NADP are used and where Rubisco functions. Describe these steps. Concept 10.5 Alternative mechanisms of carbon fixation have evolved in hot, arid climates, pp. 203. 206. On dry, hot days, plants close their stomata, conserving water but keeping out and in. Under these conditions, photorespiration can occur. Rubisco binds instead of consuming ADP and releasing without producing ADP or carbohydrate. Photorespiration may be an evolutionary relic, and it may play a photoprotective role. NCAM plants are adapted to hot, dry climates. Plants minimize the cost of photorespiration by incorporating into 4-carbon compounds in mesophyll cells. These compounds are exported to bundle sheath cells, where they release carbon dioxide for use in the Calvin cycle. CAM plants open their stomata at night, incorporating into organic acids, which are stored in mesophyll cells. During the day, the stomata close, and the is released from the organic acids for use in the Calvin cycle. Organic compounds produced by photosynthesis provide the energy and building material for Earth's ecosystems. Why are NCAM photosynthesis more energetically expensive than photosynthesis? What climate conditions would favor NCAM plants? Concept 10.6 Photosynthesis is essential for life on Earth, a review, pp. 206. 209. Organic compounds produced by photosynthesis provide the energy and building material for Earth's ecosystems. How do plants use the products of photosynthesis? Test your understanding. For more multiple choice questions, go to the practice test in the Mastering Biology e-text or study area, or go to goo.gl/gruwrg. Levels 1 to 2, Remembering/Understanding. The light reactions supply the Calvin cycle with light energy, CO2, and ADP, H2O, and NADP. ADP and NADP. Which of the following sequences correctly represents the flow of electrons during photosynthesis? NADP rightward arrow O2 rightward arrow CO2. How is photosynthesis similar in plants and CAM plants? In both cases, only photosystem I is used. Both types of plants make sugar without the Calvin cycle. In both cases, rubisco is not used to fix carbon initially. Both types of plants make most of their sugar in the dark. Which of the following statements is a correct distinction between autotrophs and heterotrophs? Autotrophs, but not heterotrophs, can nourish themselves beginning with and other nutrients that are inorganic. Only heterotrophs require chemical compounds from the environment. Cellular respiration is unique to heterotrophs. Only heterotrophs have mitochondria. Which of the following occurs during the Calvin cycle? Carbon fixation. Reduction of. Release of oxygen. Generation of. Levels 3 to 4, Applying/Analyzing. In mechanism, photophosphorylation is most similar to substrate level phosphorylation in glycolysis, oxidative phosphorylation in cellular respiration, carbon fixation, reduction of. Which process is most directly driven by light energy? Creation of a pH gradient by pumping protons across the thylakoid membrane. Reduction of molecules. Transfer of energy from pigment molecule to pigment molecule. ADP synthesis. Levels 5 to 6, Evaluating/Creating. Science, technology, and society. Scientific evidence indicates that the added to the air by the burning of wood and fossil fuels is contributing to global warming, a rise in global temperature. Tropical rainforests are estimated to be responsible for approximately 20% of global photosynthesis, yet the consumption of large amounts of by living trees is thought to make little or no net contribution to reduction of global warming. Explain why this might be the case. Hint: what processes in both living and dead trees produce evolution connection. Photorespiration can decrease soybeans' photosynthetic output by about 50%. Would this figure be higher or lower in wild relatives of soybeans? Why? Scientific inquiry. Draw it. The following diagram represents an experiment with isolated thylakoids. The thylakoids were first made acidic by soaking them in a solution at pH 4. After the thylakoid space reached pH 4, the thylakoids were transferred to a basic solution at pH 8. The thylakoids then made ADP in the dark. See Concept 3.3 to review pH. Diagram of an experiment with isolated thylakoids. 10.3 to 12 Full alternative text. Draw an enlargement of part of the thylakoid membrane in the beaker with the solution at pH 8. Draw ADP synthase. Label the areas of high concentration and low concentration. Show the direction protons flow through the enzyme, and show the reaction where ADP is synthesized. Would ADP end up in the thylakoid or outside of it? Explain why the thylakoids in the experiment were able to make ADP in the dark. Write about a theme, energy and matter in a short essay, 100 to 150 words. Describe how photosynthesis transforms solar energy into the chemical energy of sugar molecules. Synthesize your knowledge. A photo of the landscape in Antarctica. Watermelon snow in Antarctica is caused by a certain species of photosynthetic green algae that thrives in sub-zero temperatures, Chlamydomonas nivalis. These algae are also found in high-altitude, year-round snow fields. In both locations, UV light levels tend to be high. Propose an explanation for why this alga appears reddish-pink. For selected answers, see Appendix A. Explore scientific papers with science in the classroom AAAS. Can we capture the sun's energy by mimicking plants? Go to Replicating Photosynthesis at www.scienceintheclassroom.org. Instructors, questions can be assigned in Mastering Biology. Cell communication. Figure 11.1. A cheetah chases an impala. This impala is fleeing for its life, racing to escape the predatory cheetah nipping at its heels. The impala is breathing rapidly, its heart pounding and its legs pumping furiously. These physiological functions are all part of the impala's fight or flight response, driven by hormones released from its adrenal glands at times of stress, in this case, upon sensing the cheetah. How does cell signaling fuel the desperate flight of an impala? 11.1 to 1 Full alternative text. Key concepts. 11.1 External signals are converted to responses within the cell. 213. 11.2 Signal reception. A signaling molecule binds to a receptor, causing it to change shape. 217. 11.3 Signal transduction. Cascades of molecular interactions transmit signals from receptors to relay molecules in the cell. 221. 11.4 Cellular response. Cell signaling leads to regulation of transcription or cytoplasmic activity. 226. 11.5 Apoptosis requires integration of multiple cell signaling pathways. 229. Study tip. Make a table. As you read about examples of cell signaling in this chapter, make a table and classify the events of each example into 3 stages: signal reception, signal transduction, and cellular response. A table. 11.1 to 2 Full alternative text. Go to Mastering Biology. For students, in e-text and study area. Get ready for Chapter 11. Animation. Overview of cell signaling. Animation, signal transduction pathways. For instructors to assign, in item library. Problem-solving exercise, can a skin wound turn deadly? Tutorial, cell signaling, transduction and response. Concept 11.1 External signals are converted to responses within the cell. Scientists think that signaling mechanisms first evolved hundreds of millions of years ago in ancient prokaryotes and single-celled eukaryotes and then were adapted for new uses in their multicellular descendants. So let's begin by considering signaling in some examples of single-celled organisms, bacteria and yeasts. Evolution of cell signaling. Evolution research during the 1970s suggested that bacterial cells, somewhat surprisingly, since they are single-celled organisms, were capable of signaling to each other. Since then, we have come to understand that cell signaling is critical among prokaryotes. Bacterial cells secrete molecules that can be detected by other bacterial cells. Figure 11.2. Sensing the concentration of such signaling molecules allows bacteria to monitor their own local cell density, a phenomenon called quorum sensing. Figure 11.2. Communication among bacteria. Communication among bacteria. Step 1. Individual rod-shaped cells on a 0.5 mm scale signal. Step 2. Aggregation in progress. Step 3. Spore-forming structure, fruiting body, is formed on a 2.5 mm scale. Soil-dwelling bacteria called myxobacteria, slime bacteria, use chemical signals to share information about nutrient availability. When food is scarce, starving cells secrete a signaling molecule that stimulates neighboring cells to aggregate. The cells form a structure called a fruiting body that produces spores, thick-walled cells that can survive until the environment improves. The myxobacteria shown here are the species Myxococcus xanthus, steps 1 to 3, SEMS, lower photo, LM. Quorum sensing allows bacterial populations to coordinate the behavior of all cells in a population in activities that require a given density of cells acting at the same time. One example is formation of a biofilm, an aggregation of bacterial cells attached to a surface by molecules secreted by the cells, but only after the cells have reached a certain density. The biofilm protects the cells in it, and they often derive nutrition from the surface they are on. Biofilms are believed to be involved in up to 80% of all human bacterial infections. You have probably encountered biofilms many times, perhaps without realizing it. The slimy coating on a fallen log or on leaves lying on a forest path, and even the film on your teeth each morning, are examples of bacterial biofilms. In fact, toothbrushing and flossing disrupt biofilms that would otherwise cause cavities and gum disease. Another example of bacterial behavior coordinated by quorum sensing is the secretion of toxins by infectious bacteria, which has serious medical implications. Sometimes treatment by antibiotics doesn't work with such infections because of antibiotic resistance that has evolved in a particular strain of bacteria. A promising alternative treatment would be to disrupt toxin production by interfering with the signaling pathways used in quorum sensing. In the problem-solving exercise, you can participate in the process of scientific thinking involved in this novel approach. Now let's look at an example of cell signaling in yeasts, single-celled fungi. Cells of the yeast Saccharomyces cerevisiae, which are used to make bread, wine, and beer, identify their sexual mates by chemical signaling when they reproduce sexually. There are two sexes, or mating types, called A and Figure 11.3. Each type secretes a specific factor that binds only to receptors on the other type of cell. When exposed to each other's mating factors, a pair of cells of opposite type change shape, grow toward each other, and fuse, mate. The new cell contains all the genes of both original cells, providing advantages to the cell's descendants, which arise by subsequent cell divisions. Figure 11.3. Communication between mating yeast cells. Communication between yeast cells. Saccharomyces cerevisiae cells use chemical signaling to identify cells of the opposite mating type and initiate the mating process. The two mating types and their corresponding chemical signaling molecules, or mating factors, are called A and Figure 11.3. Full alternative text. Problem-solving exercise. A chart asks, can a skin wound turn deadly? 11.1 to 3. Full alternative text. Cells sense their own population density by quorum sensing, and at a certain density they start to secrete toxin. In this exercise, you will analyze whether blocking quorum sensing can stop S. aureus from producing toxin. Your approach in S. aureus, quorum sensing, involves two separate signal transduction pathways. Two candidate synthetic peptides, short proteins, called peptides 1 and 2, have been proposed to interfere with S. aureus quorum sensing pathways. Your job is to test each potential inhibitor of quorum sensing to see if it blocks either or both of the pathways that lead to toxin production. For your experiment, you grow 4 cultures of S. aureus to a standardized high density and measure the concentration of toxin in the culture. The control culture contains no peptide. The other cultures have one or both candidate inhibitory peptides mixed into the growth medium before starting the cultures. Your data. Bar graph. Y-axis is concentration of toxin in culture, micromoles per milliliter, dot. X-axis shows concentrations for 4 cultures: control, peptide 1, peptide 2, peptides 1 plus 2. Peptides 1 plus 2 shows the lowest concentration. Data from N-Balaban et al., treatment of Staphylococcus aureus biofilm infection by the quorum sensing inhibitor RIP, antimicrobial agents, and chemotherapy 51, 6/22/26, 22/29, 2007. 11.1 to 4 Full alternative text. Your analysis. Rank the cultures according to toxin production, from most to least. Which, if any, of the cultures with peptide S resulted in a toxin concentration similar to the control culture? What is your evidence for this? Was there an additive effect on toxin production when peptides 1 and 2 were both present in the growth medium? What is your evidence for this? Based on these data, would you hypothesize that peptides 1 and 2 act on the same quorum sensing pathway leading to toxin production or on 2 different pathways? What is your reasoning? Do these data suggest a possible treatment for antibiotic-resistant S. aureus infections? What else would you want to know to investigate this further? Instructors, a version of this problem-solving exercise can be assigned in Mastering Biology. Or a more extensive investigation called Solve It, Is It Possible to Treat Bacterial Infections Without Traditional Antibiotics, can be assigned. Mastering Biology. HHMI video, interview with Bonnie Bassler HHMI. BBC video, brushing your teeth can save your life. Mastering Biology. Interview with Bonnie Bassler, exploring how bacteria communicate with each other. A photo of Bonnie Bassler. Mastering Biology. The unique match between mating factor and receptor is key to ensuring mating only between cells of the same species of yeast. How does the binding of a mating factor by the yeast cell surface receptor initiate a signal that brings about the cellular response of mating? This occurs in a series of 3 major steps: signal reception, signal transduction, and cellular response, called a signal transduction pathway. Many such pathways exist in the cells of both unicellular and multicellular organisms. In fact, the molecular details of signal transduction in yeasts and mammals are strikingly similar, even though it's been over 1 billion years since they shared a common ancestor. This similarity suggests that early versions of cell signaling mechanisms evolved well before the first multicellular organisms appeared on Earth. Local and long-distance signaling. Like bacteria or yeast cells, cells in a multicellular organism communicate by way of a signaling molecules targeted for cells that may or may not be immediately adjacent. As we saw in Concept 6.7 and 7.1, eukaryotic cells may communicate by direct contact, which is one type of local signaling. Many animal and plant cells have cell junctions that directly connect the cytoplasms of adjacent cells. Figure 11.4a. In these cases, signaling substances dissolved in the cytosol can pass between neighboring cells. Moreover, some animal cells may communicate by direct contact between cell surface molecules, as shown in Figure 11.4b. This type of local signaling is especially important in embryonic development, the immune response, and in maintaining adult stem cell populations. Figure 11.4 Communication requiring contact between cells. Communication requiring contact between cells. Figure 11.4 Full alternative text. In many other cases of local signaling, signaling molecules are secreted by the signaling cell. Some molecules travel only short distances. Such local regulators influence cells that are nearby. This type of local signaling in animals is called paracrine signaling. Figure 11.5a. One class of local regulators in animals, growth factors, consists of compounds that stimulate nearby target cells to grow and divide. Numerous cells can simultaneously receive and respond to the growth factors produced by a single cell in their vicinity. Figure 11.5 Local and long-distance cell signaling by secreted molecules in animals. Local and long-distance cell signaling by secreted molecules in animals. In both local and long-distance signaling, only specific target cells that can recognize a given signaling molecule will respond to it. Figure 11.5 Full alternative text. A highly specialized type of local signaling called synaptic signaling occurs in the animal nervous system. Figure 11.5b, c. Concept 48.4. An electrical signal along a nerve cell triggers the secretion of neurotransmitter molecules. These molecules act as chemical signals, diffusing across the synapse, the narrow space between the nerve cell and its target cell, triggering a response in the target cell. Drugs to treat depression, anxiety, and post-traumatic stress disorder, PTSD, affect this signaling process. Both animals and plants use molecules called hormones for long-distance signaling. In hormonal signaling in animals, also known as endocrine signaling, specialized cells release hormones, which travel through the circulatory system to other parts of the body, where they reach target cells that can recognize and respond to them. Figure 11.5c. Many plant hormones reach distant targets by traveling through cells, see Concept 39.2. Like local regulators, plant hormones vary widely in size and type. For instance, the plant hormone ethylene, which promotes fruit ripening and helps regulate growth, is a hydrocarbon of only 6 atoms, small enough to diffuse through the air and pass through cell walls. Small signaling molecules probably evolved early on among single-celled organisms. In contrast, the mammalian hormone insulin, which regulates sugar levels in the blood, is a protein with thousands of atoms. What happens when a potential target cell is exposed to a secreted signaling molecule? The ability of a cell to respond is determined by whether it has a specific receptor molecule that can bind to the signaling molecule. The information conveyed by this binding, the signal, must then be changed into another form transduced inside the cell before the cell can respond. The remainder of the chapter discusses this process, primarily as it occurs in animal cells. 2 signaling pathway proteins are shown in their cellular context in Figure 6.32a. The 3 stages of cell signaling, a preview. Our current understanding of how signaling molecules act through signal transduction pathways had its origins in the pioneering work of Earl W. Sutherland, whose research led to a Nobel Prize in 1971. Sutherland and his colleagues at Vanderbilt University were investigating how the animal hormone epinephrine, also called adrenaline, triggers the fight or flight response in animals like the impala in Figure 11.1. One effect of epinephrine is to mobilize fuel reserves, which can be used by the animal to either defend itself, fight, or try to escape, flight. Epinephrine stimulates the breakdown of the storage polysaccharide glycogen within liver cells and skeletal muscle cells. The breakdown of glycogen releases the sugar glucose 1-phosphate, which the cell converts to glucose 6-phosphate. The liver or muscle cell can then use this compound, an early intermediate in glycolysis, for energy production, see Figure 9.8. Alternatively, the compound can be stripped of phosphate and released from the liver cell into the blood as glucose, which can fuel cells throughout the body. But how, exactly, does epinephrine mobilize glucose for use? Sutherland's research team discovered that epinephrine outside the cell stimulates glycogen breakdown by somehow activating an enzyme, glycogen phosphorylase, inside the cell. However, when epinephrine was added to a solution containing the enzyme and its substrate, glycogen, no breakdown occurred. Glycogen phosphorylase could be activated by epinephrine only when the hormone was added to intact cells. This result told Sutherland 2 things. First, epinephrine does not interact directly with the enzyme responsible for glycogen breakdown, an intermediate step or series of steps must be occurring in the cell. Second, an intact, membrane-bound cell must be present for transmission of the signal to take place. Sutherland's work suggested that the process going on at the receiving end of a cellular communication can be dissected into 3 stages: signal reception, signal transduction, and cellular response. Figure 11.6. Signal reception. Reception is the target cell's detection of a signaling molecule coming from outside the cell. A chemical signal is detected when the signaling molecule binds to a receptor protein located at the cell's surface, or inside the cell, to be discussed later. Signal transduction. The binding of the signaling molecule changes the receptor protein in some way, initiating the process of transduction. The transduction stage converts the signal to a form that can bring about a specific cellular response. In Sutherland's system, the binding of epinephrine to a receptor protein in a liver cell's plasma membrane leads to activation of glycogen phosphorylase in the cytosol. Transduction sometimes occurs in a single step but more often requires a sequence of changes in a series of different molecules. A signal transduction pathway. The molecules in the pathway are often called relay molecules. 3 are shown as an example. Cellular response. The transduced signal finally triggers a specific cellular response. The response may be almost any imaginable cellular activity such as catalysis by an enzyme, for example, glycogen phosphorylase, rearrangement of the cytoskeleton, or activation of specific genes in the nucleus. The cell signaling process helps ensure that crucial activities like these occur in the right cells, at the right time, and in proper coordination with the activities of other cells of the organism. We'll now explore the mechanisms of cell signaling in more detail, including a discussion of regulation and termination of the process. Figure 11.6 Overview of cell signaling. An overview of cell signaling. From the perspective of the cell receiving the message, cell signaling can be divided into three stages: signal reception, signal transduction, and cellular response. When reception occurs at the plasma membrane, as shown here, the transduction stage is usually a pathway of several steps. 3 are shown as an example, with each specific relay molecule in the pathway bringing about a change in the next molecule. The final molecule in the pathway triggers the cell's response. Figure 11.6 Full alternative text. Visual skills. Where would the epinephrine in Sutherland's experiment fit into this diagram of cell signaling? Mastering biology. Animation, overview of cell signaling. Concept check 11.1. Explain how signaling is involved in ensuring that yeast cells fuse only with cells of the opposite mating type. In liver cells, glycogen phosphorylase acts in which of the three stages of the signaling pathway associated with an epinephrine-initiated signal? What if epinephrine were mixed with glycogen phosphorylase and glycogen in a cell-free mixture in a test tube, would glucose 1-phosphate be generated? Why or why not? For suggested answers, see Appendix A. Concept 11.2 Signal reception. A signaling molecule binds to a receptor, causing it to change shape. A wireless router may broadcast its network signal indiscriminately, but only computers with the correct password can connect to it. Reception of the signal depends on the receiver. Similarly, the signals emitted by an mating type yeast cell are heard only by its prospective mates. Alpha cells. In the case of the epinephrine circulating throughout the bloodstream of the impala in Figure 11.1, the hormone encounters many types of cells, but only certain target cells, those with the corresponding receptor protein, detect and respond to the epinephrine molecule. The signaling molecule is complementary in shape to a specific site on the receptor and attaches there, like a hand in a glove. The signaling molecule acts as a ligand, the term for a molecule that specifically binds to another, often larger, molecule. Ligand binding generally causes a receptor protein to undergo a change in shape. For many receptors, This shape change directly activates the receptor, enabling it to interact with other molecules in or on the cell. For other receptors, the immediate effect of ligand binding is to cause the aggregation of two or more receptor proteins, which leads to further molecular events inside the cell. Most signal receptors are plasma membrane proteins, but others are located inside the cell. We'll look at both of these types next. Mastering biology. Animation, reception. Receptors in the plasma membrane. Cell surface transmembrane receptors play crucial roles in the biological systems of animals. The largest family of human cell surface receptors is that of the G-protein coupled receptors, GPCRs. There are more than 800 GPCRs, an example is shown in Figure 11.7. Another example is the co-receptor hijacked by HIV to enter immune cells, see Figure 7.8. This GPCR is the target of the drug Maraviroc, which has shown some success at treating AIDS. Figure 11.7. The structure of a G-protein coupled receptor, GPCR. A computer model of the human beta-2 adrenergic receptor within the plasma membrane. Within it is cholesterol and a molecule that mimics a ligand. This is a model of the human adrenergic receptor, which binds epinephrine, adrenaline. The receptor was crystallized, discussed later in the section, in the presence of both a molecule that mimics epinephrine, green in the model, and cholesterol in the membrane, orange. Two receptors, blue, are shown as ribbon models in a side view. Caffeine can also bind to this receptor, see question 10 at the end of the chapter. Most water-soluble signaling molecules bind to specific sites on transmembrane receptor proteins that transmit information from the extracellular environment to the inside of the cell. We can see how cell surface transmembrane receptors work by looking at three major types: G-protein coupled receptors, GPCRs, receptor tyrosine kinases, and ion channel receptors. These receptors are discussed and illustrated in Figure 11.8. Study this figure before going on. Figure 11.8. Exploring cell surface transmembrane receptors. A diagram of G-protein coupled receptors. 11.2-6 Full alternative text. A diagram of receptor tyrosine kinases. 11.2-7 Full alternative text. A diagram of ion channel receptors. 11.2-8 Full alternative text. Make connections is the flow of ions through a ligand-gated channel, an example of active or passive transport. Review. Concepts 7.3 and 7.4. Mastering biology. Animation, acetylcholine receptor. Given the many important functions of cell surface receptors, it is not surprising that their malfunctions are associated with many human diseases, including cancer, heart disease, and asthma. To better understand and treat these conditions, a major focus of both university research teams and the pharmaceutical industry has been to analyze the structure of these receptors. Although cell surface receptors, half of which are GPCRs, represent 30% of all human proteins, determining their structures by X-ray crystallography, see Figure 5.21, has proved challenging. For one thing, cell surface receptors tend to be flexible and inherently unstable, thus difficult to crystallize. It took years of persistent efforts for researchers to determine the first few of these structures, such as the GPCR shown in Figure 11.7. In that case, the adrenergic receptor was stable enough to be crystallized only while it was among membrane molecules and in the presence of a molecule mimicking its ligand. Newer techniques that do not require crystallization, like cryo-EM, see Figure 6.3, have shown promise in determining the structures of cell surface receptors. Abnormal functioning of receptor tyrosine kinases, RTKs, is associated with many types of cancers. For example, some breast cancer patients have tumor cells with excessive levels of a receptor tyrosine kinase called HER2, see Concept 12.3 and Figure 18.27. Using molecular biological techniques, researchers have developed a protein called Herceptin that binds to HER2 on cells and inhibits cell division, thus thwarting further tumor development. In some clinical studies, treatment with Herceptin improved patient survival rates by more than 1/3. One goal of ongoing research into these cell surface receptors and other cell signaling proteins is development of additional successful treatments. Mastering biology. Interview with Diana Bautista, How Cell Signaling Leads to Itch and Pain, see the interview before. Chapter 6. A photo of Diana Bautista. Intracellular receptors. Intracellular receptor proteins are found in either the cytoplasm or nucleus of target cells. To reach such a receptor, a signaling molecule passes through the target cell's plasma membrane. A number of important signaling molecules can do this because they are either hydrophobic enough or small enough to cross the hydrophobic interior of the membrane. See Concept 7.1. Hydrophobic signaling molecules include steroid hormones and thyroid hormones of animals. Another chemical signaling molecule that possesses an intracellular receptor is nitric oxide, NO, a gas. This very small molecule readily passes between the membrane phospholipids. Once a hormone or other signaling molecule has entered a cell, its binding to an intracellular receptor changes the receptor into a hormone receptor complex that is able to cause a response in many cases, the turning on or off of particular genes. The behavior of aldosterone is a representative example of how steroid hormones work. This hormone is secreted by cells of the adrenal gland, a gland that lies above the kidney, then travels through the blood and enters cells all over the body. However, a response occurs only in kidney cells because they alone contain receptors for aldosterone. In these cells, the hormone binds to and activates the receptor protein. With aldosterone attached, the active form of the receptor protein then enters the nucleus and turns on specific genes that control water and sodium flow in kidney cells, ultimately affecting blood volume. Figure 11.9. Figure 11.9 Steroid hormone interacting with an intracellular receptor. The process of steroid hormone interaction in 5 steps. Figure 11.9 Full alternative text. Make connections. Why is a cell surface receptor protein not required for the steroid hormone to enter the cell? See Concept 7.2. Mastering biology. Animation, steroid hormone pathway. How does the activated hormone receptor complex turn on genes? Recall that the genes in a cell's DNA function by being transcribed and processed into messenger RNA, mRNA, which leaves the nucleus and is translated into a specific protein by ribosomes in the cytoplasm. See Figure 5.22. Special proteins called transcription factors control which genes are turned on, that is, which genes are transcribed into mRNA in a particular cell at a particular time. When the aldosterone receptor is activated, it acts as a transcription factor that turns on specific genes. You'll learn more about transcription factors in Chapters 17 and 18. By acting as a transcription factor, the aldosterone receptor itself carries out 2 parts of the signaling pathway as receptor and transducer. Most other intracellular receptors function in the same way, although many of them, such as the thyroid hormone receptor, are already in the nucleus before the signaling molecule reaches them. Interestingly, many of these intracellular receptor proteins are structurally similar, suggesting an evolutionary kinship. Concept check 11.2. Nerve growth factor, NGF, is a water-soluble signaling molecule. Would you expect the receptor for NGF to be intracellular or in the plasma membrane? Explain. What if? What would the effect be if a cell made defective receptor tyrosine kinase proteins that were unable to dimerize? Make connections. How is ligand binding similar to the process of allosteric regulation of enzymes? See Figure 8.20. For suggested answers, see Appendix A. Concept 11.3 Signal transduction. Cascades of molecular interactions transmit signals from receptors to relay molecules in the cell. When receptors for signaling molecules are plasma membrane proteins, like most of those we have discussed, the transduction stage of cell signaling is usually a multi-step pathway involving many molecules. Steps often include activation of proteins by addition or removal of phosphate groups or release of other small molecules or ions that act as signaling molecules. One benefit of using multiple steps is that a signal caused by a small number of signaling molecules can be greatly amplified. If each molecule transmits the signal to numerous molecules at the next step in the series, the result is a geometric increase in the number of activated molecules by the end. See Figure 11.16. A second benefit of using multi-step pathways is that they provide more opportunities for coordination and control than do simpler systems. This allows regulation of the response, as we'll see later in the chapter. Signal transduction pathways. The binding of a specific signaling molecule to a receptor in the plasma membrane triggers the first step in the signal transduction pathway, the chain of molecular interactions that leads to a particular response within the cell. Like falling dominoes, the signal-activated receptor activates another molecule, which activates yet another molecule, and so on, until the protein that produces the final cellular response is activated. The molecules that relay a signal from receptor to response, which we call relay molecules in this book, are often proteins. Protein-protein interactions are a major theme of cell signaling, indeed, a unifying theme of all cellular activities. Mastering biology. Animation, signal transduction pathways. Keep in mind that the original signaling molecule is not physically passed along a signaling pathway. In most cases, it never even enters the cell. When we say that the signal is relayed along a pathway, we mean that certain information is passed on. At each step, the signal is transduced into a different form, commonly a shape change in the next protein. Very often, the shape change is brought about by phosphorylation. Protein phosphorylation and dephosphorylation. Previous chapters introduced the concept of activating a protein by adding one or more phosphate groups to it. See Figure 8.11a. In Figure 11.8, you have already seen how phosphorylation is involved in the activation of receptor tyrosine kinases. In fact, phosphorylation of proteins and its reverse, dephosphorylation, are commonly used in cells to regulate protein activity. An enzyme that transfers phosphate groups from ADP to a protein is generally known as a protein kinase. Recall that a receptor tyrosine kinase, RTK, is a specific kind of protein kinase that phosphorylates tyrosines on the other RTK in a dimer. Most cytoplasmic protein kinases, however, act on proteins different from themselves. Another distinction is that most cytoplasmic protein kinases phosphorylate either of two other amino acids, serine or threonine, rather than tyrosine. Serine/threonine kinases are widely involved in signaling pathways in animals, plants, and fungi. Many signal transduction pathways use relay molecules that are protein kinases, and they often act on other protein kinases in the pathway. Figure 11.10 depicts a hypothetical pathway containing two different protein kinases that create a phosphorylation cascade. The sequence of steps shown in the figure is similar to many known pathways, including those triggered in yeast by mating factors and in animal cells by many growth factors. The signal is transmitted by a cascade of protein phosphorylations, each causing a shape change in the phosphorylated protein. The shape change results from the interaction of the newly added phosphate groups with charged or polar amino acids on the protein being phosphorylated. See Figure 5.14. The shape change in turn alters the function of the protein, most often activating it. In some cases, though, phosphorylation instead decreases the activity of the protein. Figure 11.10 A phosphorylation cascade. A phosphorylation cascade in 4 steps. In a phosphorylation cascade, a series of different proteins in a pathway are phosphorylated in turn, each protein adding a phosphate group to the next one in line. Here, phosphorylation activates each protein, and dephosphorylation returns it to its inactive form. The active and inactive forms of each protein are represented by different shapes to remind you that activation is usually associated with a change in molecular shape. Figure 11.10 Full alternative text. What if? What would happen if a mutation in protein kinase 2 made it incapable of being phosphorylated? A significant percentage of our own genes, about 2%, is thought to code for protein kinases. A single cell may have hundreds of different kinds, each specific for a different substrate protein. Together, protein kinases probably regulate the activity of a large proportion of the thousands of proteins in a cell. Among these are most of the proteins that, in turn, regulate cell division. Abnormal activity of such a kinase can cause abnormal cell division and contribute to the development of cancer. Equally important in the phosphorylation cascade are the protein phosphatases, enzymes that can rapidly remove phosphate groups from proteins, a process called dephosphorylation. By dephosphorylating and thus inactivating protein kinases, phosphatases provide the mechanism for turning off the signal transduction pathway when the initial signal is no longer present. Phosphatases also make the protein kinases available for reuse, enabling the cell to respond again to an extracellular signal. The phosphorylation-dephosphorylation system acts as a molecular switch in the cell, turning activities on or off, or up or down, as required. At any given moment, the activity of a protein regulated by phosphorylation depends on the balance in the cell between active kinase molecules and active phosphatase molecules. Small molecules and ions as second messengers. Not all components of signal transduction pathways are proteins. Many signaling pathways also involve small, non-protein, water-soluble molecules or ions called second messengers. The pathway's first messenger is considered to be the extracellular signaling molecule, the ligand that binds to the membrane receptor. Because they are small and water-soluble, they can readily spread through regions of the cell by diffusion. For example, as we'll see shortly, a second messenger called cyclic AMP carries the signal initiated by epinephrine from the plasma membrane of a liver or muscle cell into the cell's interior, where the signal eventually brings about glycogen breakdown. Second messengers participate in pathways that are initiated by both G-protein coupled receptors and receptor tyrosine kinases. The two most common second messengers are cyclic AMP and calcium ions. A large variety of relay proteins respond to changes in the cytosolic concentration of one or the other of these second messengers. Cyclic AMP. Having discovered that epinephrine somehow causes glycogen breakdown within cells, Earl Sutherland next looked for a second messenger that transmits the signal from the plasma membrane to the metabolic machinery in the cytoplasm. He found that binding of epinephrine to the G-protein coupled receptor, GPCR, in the plasma membrane results in a rise in the cytosolic concentration of cyclic AMP, CAMP, cyclic adenosine monophosphate, a small molecule produced from ADP. As shown in Figure 11.11, an enzyme embedded in the plasma membrane. Adenylyl cyclase, also known as adenylate cyclase, converts ADP to CAMP in response to an extracellular signal, in this case, provided by epinephrine. When epinephrine outside the cell binds to a GPCR, it activates a G protein that in turn activates adenylyl cyclase. Adenylyl cyclase can then catalyze the synthesis of many molecules of CAMP. In this way, the normal cellular concentration of CAMP can be boosted 20-fold in a matter of seconds. The CAMP broadcasts the signal to the cytoplasm. It does not persist for long in the absence of the hormone because a different enzyme, called phosphodiesterase, converts CAMP to AMP. Another surge of epinephrine is needed to boost the cytosolic concentration of CAMP again. Figure 11.11 Cyclic AMP. Cyclic AMP. The second messenger cyclic AMP, CAMP, is made from ADP by adenylyl cyclase, an enzyme embedded in the plasma membrane. The phosphate group in CAMP is attached to both the and the carbons. CAMP cyclic arrangement is the basis for its name. CAMP is inactivated by phosphodiesterase, an enzyme that converts it to AMP. Figure 11.11 Full alternative text. What if? What would happen if a molecule that inactivated phosphodiesterase were introduced into the cell? Subsequent research has revealed that epinephrine is only one of many hormones and many other signaling molecules that lead to activation of adenylyl cyclase by G proteins and formation of CAMP. Figure 11.12. The immediate effect of an elevation in CAMP level is usually the activation of a serine/threonine kinase called protein kinase A. The activated protein kinase A then phosphorylates various other proteins, depending on the cell type. The complete pathway for epinephrine stimulation of glycogen breakdown is shown later, in Figure 11.16. Figure 11.12 CAMP as a second messenger in a G protein signaling pathway. CAMP as a second messenger in a G protein signaling pathway. Figure 11.12 Full alternative text. Draw it. The bacterium that causes the disease cholera produces a toxin that locks the G protein in its activated state. Review. Figure 11.8, then draw this figure as it would be if cholera toxin were present. You do not need to draw the cholera toxin molecule. Further regulation of cell metabolism is provided by other G protein systems that inhibit adenylyl cyclase. In these systems, a different signaling molecule activates a different receptor, which in turn activates an inhibitory G protein that blocks activation of adenylyl cyclase. Cell activities can be fine-tuned by the balance between these systems. Now that we know about the role of CAMP in G-protein signaling pathways, we can explain in molecular detail how certain microorganisms cause disease. Consider cholera, a disease that is frequently epidemic in places where the water supply is contaminated with human feces. People acquire the cholera bacterium, Vibrio cholerae, by drinking contaminated water. The bacteria form a biofilm on the lining of the small intestine and produce a toxin. The cholera toxin is an enzyme that chemically modifies a G protein involved in regulating salt and water secretion. Because the modified G protein is unable to hydrolyze GTP to GDP, it remains stuck in its active form, continuously stimulating adenylyl cyclase to make CAMP. See the question with Figure 11.12. The resulting high concentration of CAMP causes the intestinal cells to secrete large amounts of salts into the intestines, with water following by osmosis. An infected person quickly develops profuse diarrhea and if left untreated can soon die from the loss of water and salts. Our understanding of signaling pathways involving cyclic AMP or related messengers has allowed us to develop treatments for certain conditions in humans such as erectile dysfunction. In one pathway, the gas nitric oxide, NO, is released by a cell and enters a neighboring muscle cell, where it causes production of a molecule similar to CAMP called cyclic GMP, cGMP. The cGMP then acts as a second messenger that causes relaxation of muscles, such as those in the walls of arteries. A compound that prolongs the signal, by inhibiting the hydrolysis of cGMP to GMP, was originally prescribed for chest pains because it relaxed blood vessels and increased blood flow to the heart muscle. Under the trade name Viagra, this compound is now widely used as a treatment for erectile dysfunction in human males. Because Viagra leads to dilation of blood vessels, it also allows increased blood flow to the penis, optimizing physiological conditions for penile erections. Calcium ions and inositol trisphosphate. Many of the signaling molecules that function in animals including neurotransmitters, growth factors, and some hormones induce responses in their target cells through signal transduction pathways that increase the cytosolic concentration of calcium ions. Calcium is even more widely used than CAMP as a second messenger. Increasing the local cytosolic concentration of causes many responses in animal cells, including muscle cell contraction, exocytosis of molecules, secretion, and cell division. In plant cells, a wide range of hormonal and environmental stimuli can cause brief increases in cytosolic concentration, triggering various signaling pathways, such as the pathway for greening in response to light. See Figure 39.4. Cells use as a second messenger in pathways triggered by both G-protein coupled receptors and receptor tyrosine kinases. Although cells always contain some, this ion can function as a second messenger because its concentration in the cytosol is normally much lower than the concentration outside the cell. Figure 11.13. In fact, the level of in the blood and extracellular fluid of an animal is often more than 10,000 times higher than that in the cytosol. Calcium ions are actively transported out of the cell and are actively imported from the cytosol into the endoplasmic reticulum, and, under some conditions, into mitochondria and chloroplasts, by various protein pumps. As a result, the calcium concentration in the ER is usually much higher than that in the cytosol. Because the cytosolic calcium level is low, a small change in absolute numbers of ions represents a relatively large percentage change in local calcium concentration. Figure 11.13. The maintenance of calcium ion concentrations in an animal cell. The maintenance of calcium ion concentrations in an animal cell. Ca2+ pumps are in the plasma membrane, mitochondrion membrane, and ER. ATP drives these pumps. The concentration in the cytosol is usually much lower, light green, than in the extracellular fluid and ER, dark green. Protein pumps in the plasma membrane and the ER membrane, driven by ATP, move from the cytosol into the extracellular fluid and into the lumen of the ER. Mitochondrial pumps, driven by chemiosmosis, see Concept 9.4, move into mitochondria when the calcium level in the cytosol rises significantly. In response to a signal relayed by a signal transduction pathway, the cytosolic calcium level may rise, usually by a mechanism that releases from the cell's ER. The pathways leading to calcium release involve two other second messengers: inositol trisphosphate and diacylglycerol, DAG. These two messengers are produced by cleavage of a certain kind of phospholipid in the plasma membrane. Figure 11.14 shows the complete picture of how a signal causes to stimulate the release of calcium from the ER. Because acts before calcium in these pathways, calcium could be considered a third messenger. However, scientists use the term second messenger for all small, non-protein components of signal transduction pathways. Figure 11.14 Calcium and in signaling pathways. Calcium and IP3 in signaling pathways. Calcium ions and inositol trisphosphate function as second messengers in many signal transduction pathways. In this figure, the process is initiated by the binding of a signaling molecule to a G protein-coupled receptor. A receptor tyrosine kinase could also initiate this pathway by activating phospholipase C. Figure 11.14 Full alternative text. Make connections explain the difference in function, in terms of ion transport, between the pump-in. Figure 11.13 and the channel protein shown here. See. Figure 7.17. Concept check 11.3. What is a protein kinase, and what is its role in a signal transduction pathway? When a signal transduction pathway involves a phosphorylation cascade, how does the cell's response get turned off? What is the actual signal that is being transduced in any signal transduction pathway, such as those shown in Figures 11.6 and 11.10? In what way is this information being passed from the exterior to the interior of the cell? What if you exposed a cell to a ligand that binds to a receptor and activates phospholipase C, predict the effect the gated calcium channel would have on concentration in the cytosol? For suggested answers, see Appendix A. Concept 11.4 Cellular response. Cell signaling leads to regulation of transcription or cytoplasmic activities. We now take a closer look at the cell's subsequent response to an extracellular signal, what some researchers call the output response. What is the nature of the final step in a signaling pathway? Nuclear and cytoplasmic responses. Ultimately, a signal transduction pathway leads to the regulation of one or more cellular activities. The response may occur in the nucleus or in the cytoplasm of the cell. Many signaling pathways ultimately regulate protein synthesis, usually by turning specific genes on or off in the nucleus. Like an activated steroid receptor, see Figure 11.9, the final activated molecule in a signaling pathway may function as a transcription factor. Figure 11.15 shows an example in which a signaling pathway activates a transcription factor that turns a gene on. The response to this growth factor signal is transcription, the synthesis of one or more specific mRNAs, which will be translated in the cytoplasm into specific proteins. In other cases, the transcription factor might regulate a gene by turning it off. Often, a transcription factor regulates several different genes. Figure 11.15 Nuclear responses to a signal, the activation of a specific gene by a growth factor. Reception, transduction, and response in the form of mRNA transcription illustrated in reaction to the binding of a growth factor to a receptor in the plasma membrane. This diagram shows a typical signaling pathway that leads to regulation of gene activity in the cell nucleus. The initial signaling molecule, in this case a growth factor, triggers a phosphorylation cascade, as in Figure 11.10. The ATP molecules and phosphate groups are not shown. Once phosphorylated, the last kinase in the sequence enters the nucleus and activates a transcription factor, which stimulates transcription of a specific gene, or genes. The resulting RNAs then direct the synthesis of a particular protein. Figure 11.15 Full alternative text. Mastering biology. Animation, nuclear response, activating a gene. Sometimes a signaling pathway may regulate the activity of proteins rather than causing their synthesis by activating gene expression. This directly affects proteins that function outside the nucleus. For example, a signal may cause the opening or closing of an ion channel in the plasma membrane or a change in the activity of a metabolic enzyme. As we have seen, the response of liver cells to the hormone epinephrine helps regulate cellular energy metabolism by affecting the activity of an enzyme. The final step in the epinephrine signaling pathway activates the enzyme that catalyzes the breakdown of glycogen. Figure 11.16 shows the complete pathway leading to the release of glucose 1-phosphate molecules from glycogen. Notice that as each molecule is activated, the response is amplified, a subject we'll return to shortly. Figure 11.16 Cytoplasmic response to a signal, the stimulation of glycogen breakdown by epinephrine, adrenaline. Cytoplasmic response to a signal. In this signaling system, the hormone epinephrine acts through a G-protein coupled receptor to activate a succession of relay molecules, including CAMP and 2 protein kinases, see also. Figure 11.12. The final protein activated is the enzyme glycogen phosphorylase, which uses inorganic phosphate to release glucose monomers from glycogen in the form of glucose 1-phosphate molecules. This pathway amplifies the hormonal signal. One receptor protein can activate approximately 100 molecules of G protein, and each enzyme in the pathway, once activated, can act on many molecules of its substrate, the next molecule in the cascade. The number of activated molecules given for each step is approximate. Figure 11.16 Full alternative text. Visual skills in the figure. How many glucose 1-phosphate molecules are released in response to one signaling molecule? Calculate the factor by which the response is amplified in going from each step to the next. Mastering biology. Animation, cytoplasmic response, glycogen breakdown. Signal receptors, relay molecules, and second messengers participate in a variety of pathways, leading to both nuclear and cytoplasmic responses, including cell division. Malfunctioning of growth factor pathways like the one in Figure 11.15 can contribute to abnormal cell division and the development of cancer, as we'll see in Concept 18.5. Regulation of the response. Whether the response occurs in the nucleus or in the cytoplasm, it is not simply turned on or off. Rather, the extent and specificity of the response are regulated in multiple ways. Here we'll consider 4 aspects of this regulation. First, as mentioned earlier, signaling pathways generally amplify the cell's response to a single signaling event. The degree of amplification depends on the function of the specific molecules in the pathway. Second, the many steps in a multi-step pathway provide control points at which the cell's response can be further regulated, contributing to the specificity of the response and allowing coordination with other signaling pathways. Third, the overall efficiency of the response is enhanced by the presence of proteins known as scaffolding proteins. Finally, a crucial point in regulating the response is the termination of the signal. Signal amplification. Elaborate enzyme cascades amplify the cell's response to a signal. At each catalytic step in the cascade, the number of activated products can be much greater than in the preceding step. For example, in the epinephrine-triggered pathway in Figure 11.16, each adenylyl cyclase molecule catalyzes the formation of 100 or South CAMP molecules. Each molecule of protein kinase A phosphorylates about 10 molecules of the next kinase in the pathway, and so on. The amplification effect stems from the fact that these proteins persist in their active form long enough to process multiple molecules of substrate before they become inactive again. As a result of the signal's amplification, a small number of epinephrine molecules binding to receptors on the surface of a liver cell or muscle cell can lead to the release of hundreds of millions of glucose molecules from glycogen. The specificity of cell signaling and coordination of the response. Consider 2 different cells in your body, a liver cell and a heart muscle cell, for example. Both are in contact with your bloodstream and are therefore constantly exposed to many different hormone molecules, as well as to local regulators secreted by nearby cells. Yet the liver cell responds to some signals but ignores others, and the same is true for the heart cell. And some kinds of signals trigger responses in both cells but different responses. For instance, epinephrine stimulates the liver cell to break down glycogen, but the main response of the heart cell to epinephrine is contraction, leading to a more rapid heartbeat. How do we account for this difference? The explanation for the specificity exhibited in cellular responses to signals is the same as the basic explanation for virtually all differences between cells, because different kinds of cells turn on different sets of genes, different kinds of cells have different collections of proteins. The response of a cell to a signal depends on its particular collection of signal receptor proteins, relay proteins, and proteins needed to carry out the response. A liver cell, for example, is poised to respond appropriately to epinephrine by having the proteins listed in Figure 11.16 as well as those needed to manufacture glycogen. Thus, two cells that respond differently to the same signal differ in one or more proteins that respond to the signal. Also, within some cells, there are more complex pathways that branch or converge, as shown in Figure 11.17. Notice, though, that these different pathways still have some molecules in common. For example, cells A, B, and C all use the same receptor protein for the red signaling molecule. Differences in other proteins account for their differing responses. In cell D, a different receptor protein is used for the same signaling molecule, leading to yet another response. In cell B, a pathway triggered by one signal diverges to produce two responses. Such branched pathways often involve receptor tyrosine kinases, which can activate multiple relay proteins, or second messengers, which can regulate numerous proteins. In cell C, two pathways triggered by separate signals converge to modulate a single response. Branching of pathways and crosstalk, interaction, between pathways are important in regulating and coordinating a cell's responses to information coming in from different sources in the body. You'll learn more about this coordination in Concept 11.5. Moreover, the use of some of the same proteins in more than one pathway allows the cell to economize on the number of different proteins it must make. Figure 11.17 The specificity of cell signaling. Specificity of cell signaling shown in cells A, B, C and D. The particular proteins a cell possesses determine what signaling molecules it responds to and the nature of the response. The four cells in these diagrams respond to the same signaling molecule, red, in different ways because each has a different set of proteins, purple and teal. Note, however, that the same kinds of molecules can participate in more than one pathway. Figure 11.17 Full alternative text. Visual skills study the signaling pathway shown in Figure 11.14, and explain how the situation pictured for cell B in Figure 11.17 could apply to that pathway. Instructors, the scientific skills exercise using experiments to test a model can be assigned in Mastering Biology. It explores the cellular response of a yeast cell to the signal initiated by a mating factor from a cell of the opposite mating type. Signaling efficiency, scaffolding proteins and signaling complexes. The illustrations of signaling pathways in Figure 11.17, as well as diagrams of other pathways in this chapter, are greatly simplified. The diagrams show only a few relay molecules and, for clarity's sake, display these molecules spread out in the cytosol. If this were true in the cell, signaling pathways would operate very inefficiently because most relay molecules are proteins, and proteins are too large to diffuse quickly through the viscous cytosol. How does a given protein kinase, for instance, find its protein substrate? In many cases, the efficiency of signal transduction is apparently increased by the presence of scaffolding proteins, large relay proteins to which several other relay proteins are simultaneously attached. Figure 11.18. Researchers have found scaffolding proteins in brain cells that permanently hold together networks of signaling pathway proteins at synapses. This hardwiring enhances the speed and accuracy of signal transfer between cells because the rate of protein-protein interaction is not limited by diffusion. Furthermore, in some cases the scaffolding proteins themselves may directly activate relay proteins. Figure 11.18 A scaffolding protein. A signaling molecule is in the active site of a receptor in the plasma membrane. The receptor has three different protein kinases held together by a scaffolding protein. The scaffolding protein shown here, orange, simultaneously binds to a specific activated membrane receptor and three different protein kinases. This physical arrangement facilitates signal transduction by these molecules. The importance of the relay proteins that serve as points of branching or intersection in signaling pathways is highlighted by the problems arising when these proteins are defective or missing. For instance, in an inherited disorder called Wiskott-Aldrich syndrome, the absence of a single relay protein leads to such diverse effects as abnormal bleeding, eczema, and a predisposition to infections and leukemia. These symptoms are thought to arise primarily from the absence of the protein in cells of the immune system. By studying normal cells, scientists found that the WAS protein is located just beneath the immune cell surface. The protein interacts both with microfilaments of the cytoskeleton and with several different components of signaling pathways that relay information from the cell surface, including pathways regulating immune cell proliferation. This multifunctional relay protein is thus both a branch point and an important intersection point in a complex signal transduction network that controls immune cell behavior. When the WAS protein is absent, the cytoskeleton is not properly organized and signaling pathways are disrupted, leading to the WAS symptoms. Termination of the signal. In the interest of keeping Figure 11.17 simple, we did not show the inactivation mechanisms that are an essential aspect of any cell signaling pathway. For a cell of a multicellular organism to remain capable of responding to incoming signals, each molecular change in its signaling pathways must last only a short time. As we saw in the cholera example, if a signaling pathway component becomes locked into one state, whether active or inactive, consequences for the organism can be serious. The ability of a cell to receive new signals depends on reversibility of the changes produced by prior signals. The binding of signaling molecules to receptors is reversible. As the external concentration of signaling molecules falls, fewer receptors are bound at any given moment, and the unbound receptors revert to their inactive form. The cellular response occurs only when the concentration of receptors with bound signaling molecules is above a certain threshold. When the number of active receptors falls below that threshold, the cellular response ceases. Then, by a variety of means, the relay molecules return to their inactive forms. The GT-POS-A activity intrinsic to a G protein hydrolyzes its bound GTP, the enzyme phosphodiesterase converts CAMP to AMP, protein phosphatases inactivate phosphorylated kinases and other proteins, and so forth. As a result, the cell is soon ready to respond to a fresh signal. Mastering biology. Bioflix registered animation, mechanism of hormone action, second messenger CAMP. In this section, we explored the complexity of signaling initiation and termination in a single pathway, and we saw the potential for pathways to intersect with each other. In the next section, we'll consider one especially important network of interacting pathways in the cell. Concept check 11.4. How can a target cell's response to a single hormone molecule result in a response that affects a million other molecules? What if? If two cells have different scaffolding proteins, explain how they might behave differently in response to the same signaling molecule. What if? Some human diseases are associated with malfunctioning protein phosphatases. How would such proteins affect signaling pathways? Review the discussion of protein phosphatases in Concept 11.3 and C. Figure 11.10. Epinephrine affects heart muscle cells by causing them to mobilize glucose, contract faster, and increase heart rate. The muscle cells around lungs and airways, on the other hand, have the opposite response to epinephrine, they relax, allowing more air to be breathed in. What might explain why respiratory, breathing-related, muscle cells can respond so differently from heart muscle cells? For suggested answers, see Appendix A. Concept 11.5 Apoptosis requires integration of multiple cell signaling pathways. When signaling pathways were first discovered, they were thought to be linear, independent pathways. Our understanding of cellular communication has benefited from the realization that signaling pathway components interact with each other in various ways. For a cell to carry out the appropriate response, cellular proteins often must integrate multiple signals. Let's consider an important cellular process, cellular suicide, as an example. Cells that are infected, are damaged, or have reached the end of their functional lifespan often undergo programmed cell death. Figure 11.19. The best understood type of this controlled cell suicide is apoptosis, from the Greek, meaning falling off, and used in a classic Greek poem to refer to leaves falling from a tree. During this process, cellular agents chop up the DNA and fragment the organelles and other cytoplasmic components. The cell shrinks and becomes lobed, a change called blubbing, and the cell's parts are packaged up in vesicles that are engulfed and digested by specialized scavenger cells, leaving no trace. Apoptosis protects neighboring cells from damage that they would otherwise suffer if a dying cell merely leaked out all its contents, including its many digestive enzymes. Figure 11.19 Apoptosis of a human white blood cell. The cell on the left is more spherical while the cell on the right is covered in lobes. The scale is 2 micrometers. On the left is a normal white blood cell, while on the right is a white blood cell undergoing apoptosis. The apoptotic cell is shrinking and forming lobes, blebs, which eventually are shed as membrane-bounded cell fragments, colorized SEMs. Mastering Biology. Video, phosphate-induced apoptosis. The signal that triggers all of the complex events that occur during apoptosis can come from either outside or inside the cell. Outside the cell, signaling molecules released from other cells can initiate a signal transduction pathway that activates the genes and proteins responsible for carrying out cell death. Within a cell whose DNA has been irretrievably damaged, a series of protein-protein interactions can pass along a signal that similarly triggers cell death. Considering some examples of apoptosis can help us to see how signaling pathways are integrated in cells. Apoptosis in the soilworm Caenorhabditis elegans. The molecular mechanisms of apoptosis were worked out by researchers studying embryonic development of a small soilworm, a nematode called Caenorhabditis elegans. Because the adult worm has only about 1,000 cells, the researchers were able to work out the entire ancestry of each cell. The timely suicide of cells occurs exactly 131 times during normal development of C. elegans, at precisely the same points in the cell lineage of each worm. In worms and other species, apoptosis is triggered by signals that activate a cascade of suicide proteins in the cells destined to die. Genetic research on C. elegans initially revealed 2 key apoptosis genes, called SED3 and SED4. SED stands for cell death, which encode proteins essential for apoptosis. The proteins are called SED3 and SED4, respectively. These and most other proteins involved in apoptosis are continually present in cells, but in inactive form. Thus, regulation in this case occurs at the level of protein activity rather than through gene activity and protein synthesis. In C. elegans, a protein in the outer mitochondrial membrane, called SED9, the product of the SED9 gene, serves as a master regulator of apoptosis, acting as a brake in the absence of a signal promoting apoptosis. Figure 11.28. When a death signal is received by the cell, signal transduction involves a change in SED9 that disables the brake, and the apoptotic pathway activates proteases and nucleases, enzymes that cut up the proteins and DNA of the cell. The main proteases of apoptosis are called caspases. In the nematode, the chief caspase is the SED3 protein. Figure 11.20 Molecular basis of apoptosis in C. elegans. Molecular basis of apoptosis in C. elegans. 3 proteins, SED3, SED4, and SED9, are critical to apoptosis and its regulation in the nematode. Apoptosis is more complicated in mammals but involves proteins similar to those in C. elegans. Figure 11.20 Full alternative text. Apoptotic pathways and the signals that trigger them. In humans and other mammals, several different pathways, involving about 15 different caspases, can carry out apoptosis. The pathway that is used depends on the type of cell and on the particular signal that initiates apoptosis. One major pathway involves certain mitochondrial proteins that are triggered to form molecular pores in the mitochondrial outer membrane, causing it to leak and release other proteins that promote apoptosis. Perhaps surprisingly, these latter include cytochrome C, which functions in mitochondrial electron transport in healthy cells, see Figure 9.14, but acts as a cell death factor when released from mitochondria. The process of mitochondrial apoptosis in mammals uses proteins similar to the nematode proteins SED3, SED4, and SED9. These can be thought of as relay proteins capable of transducing the apoptotic signal. Mastering biology. Animation, apoptosis. At key gateways into the apoptotic program, relay proteins integrate signals from several different sources and can send a cell down an apoptotic pathway. Often, the signal originates outside the cell, like the death signaling molecule depicted in Figure 11.20b, which presumably was released by a neighboring cell. When a death signaling ligand occupies a cell surface receptor, this binding leads to activation of caspases and other enzymes that carry out apoptosis, without involving the mitochondrial pathway. This process of signal reception, transduction, and response is similar to what we have discussed throughout this chapter. In a twist on the classic scenario, two other types of alarm signals that can lead to apoptosis originate from inside the cell rather than from a cell surface receptor. One signal comes from the nucleus, generated when the DNA has suffered irreparable damage, and a second comes from the endoplasmic reticulum when excessive protein misfolding occurs. Mammalian cells make life or death decisions by somehow integrating the death signals and life signals they receive from these external and internal sources. A built-in cell suicide mechanism is essential to development and maintenance in all animals. The similarities between apoptosis genes in nematodes and those in mammals, as well as the observation that apoptosis occurs in multicellular fungi and even in single-celled yeasts, indicate that the basic mechanism evolved early in the evolution of eukaryotes. In vertebrates, apoptosis is essential for normal development of the nervous system, for normal operation of the immune system, and for normal morphogenesis of hands and feet in humans and paws in other mammals. Figure 11.21. The level of apoptosis between the developing digits is lower in the webbed feet of ducks and other water birds than in the non-webbed feet of land birds, such as chickens. In the case of humans, the failure of appropriate apoptosis can result in webbed fingers and toes. Figure 11.21 Effect of apoptosis during paw development in the mouse. Effect of apoptosis during paw development in the mouse, shown in 3 images. In mice, humans, other mammals, and land birds, the embryonic region that develops into feet or hands initially has a solid, plate-like structure. Apoptosis eliminates the cells in the interdigital regions, thus forming the digits. The embryonic mouse paws shown in these fluorescence light micrographs are stained so that cells undergoing apoptosis appear a bright yellowish green. Apoptosis of cells begins at the margin of each interdigital region, left, peaks as the tissue in these regions is reduced, middle, and is no longer visible when the interdigital tissue has been eliminated, right. Figure 11.21 Full alternative text. Significant evidence points to the involvement of apoptosis in certain degenerative diseases of the nervous system, such as Parkinson's disease and Alzheimer's disease. In Alzheimer's disease, an accumulation of aggregated proteins in neuronal cells activates an enzyme that triggers apoptosis, resulting in the loss of brain function seen in these patients. Furthermore, cancer can result from a failure of cell suicide. Some cases of human melanoma, for example, have been linked to faulty forms of the human version of the C. elegans SED4 protein. It is not surprising, therefore, that the signaling pathways feeding into apoptosis are quite elaborate. After all, the life or death question is the most fundamental one imaginable for a cell. This chapter has introduced you to many of the general mechanisms of cell communication, such as ligand binding, protein-protein interactions and shape changes, cascades of interactions, and protein phosphorylation. Throughout your study of biology, you will encounter numerous examples of cell signaling. Concept check 11.5. Give an example of apoptosis during embryonic development, and explain its function in the developing embryo. What if? If apoptosis occurred when it should not, what types of protein defects might be the cause? What types could result in apoptosis not occurring when it should? For suggested answers, see Appendix A. Summary of key concepts. Go to Mastering Biology for assignments, the e-text, the study area, and dynamic study modules. To review key terms, go to the vocabulary self-quiz in the Mastering Biology e-text or study area, or go to GOO.GL/ZKJZ9T. Concept 11.1 External signals are converted to responses within the cell, pp. 213. 217. Signal transduction pathways are crucial for many processes. Bacterial cells can sense the local density of bacterial cells, quorum sensing. Signaling during yeast cell mating has much in common with processes in multicellular organisms, suggesting an early evolutionary origin of signaling mechanisms. Local signaling by animal cells involves direct contact or the secretion of local regulators. For long-distance signaling, animal and plant cells use hormones. Animals also pass signals electrically. Like epinephrine, other hormones that bind to membrane receptors trigger a three-stage cell signaling pathway. An overview of cell signaling. 11.2 to 11 Full alternative text. What determines whether a cell responds to a hormone such as epinephrine? What determines how a cell responds to such a hormone? Concept 11.2 Signal reception. A signaling molecule binds to a receptor, causing it to change shape, pp. 217. 221. The binding between signaling molecule, ligand, and receptor is highly specific. A specific shape change in a receptor is often the initial transduction of the signal. Three of the major types of cell surface transmembrane receptors are the following. 1. G protein-coupled receptors, GPCRs, work with cytoplasmic G proteins. Ligand binding activates the receptor, which then activates a specific G protein, which activates yet another protein, thus propagating the signal. 2. Receptor tyrosine kinases, RTKs, react to the binding of signaling molecules by forming dimers and then adding phosphate groups to tyrosines on the cytoplasmic part of the other monomer making up the dimer. Relay proteins in the cell can then be activated by binding to different phosphorylated tyrosines, allowing this receptor to trigger several pathways at once. 3. Ligand-gated ion channels open or close in response to binding by specific signaling molecules, regulating the flow of specific ions across the membrane. The activity of all three types of receptors is crucial. Abnormal GPCRs and RTKs are associated with many human diseases. Intracellular receptors are cytoplasmic or nuclear proteins. Signaling molecules that are hydrophobic or small enough to cross the plasma membrane bind to these receptors inside the cell. How are the structures of a GPCR and an RTK similar? How does initiation of signal transduction differ for these two types of receptors? Concept 11.3 Signal transduction. Cascades of molecular interactions transmit signals from receptors to relay molecules in the cell, pp. 221. 225. At each step in a signal transduction pathway, the signal is transduced into a different form, which commonly involves a shape change in a protein. Many signal transduction pathways include phosphorylation cascades, in which a series of protein kinases each add a phosphate group to the next one in line, activating it. Enzymes called protein phosphatases remove the phosphate groups. The balance between phosphorylation and dephosphorylation regulates the activity of proteins involved in the sequential steps of a signal transduction pathway. Second messengers, such as the small molecule cyclic AMP, CAMP, and the ion diffuse readily through the cytosol and thus help broadcast signals quickly. Many G proteins activate adenylyl cyclase, which makes CAMP from ADP. Cells use as a second messenger in GPCR and RTK pathways. The tyrosine kinase pathways can also involve two other second messengers, diacylglycerol, DAG, and inositol trisphosphate. Can trigger a subsequent increase in level. What is the difference between a protein kinase and a second messenger? Can both operate in the same signal transduction pathway? Concept 11.4 Cellular response. Cell signaling leads to regulation of transcription or cytoplasmic activities, pp. 226. 229. Some pathways lead to a nuclear response. Specific genes are turned on or off by activated transcription factors. In others, the response involves cytoplasmic regulation. Cellular responses are not simply on or off, they are regulated at many steps. Each protein in a signaling pathway amplifies the signal by activating multiple copies of the next component. For long pathways, the total amplification may be over 1,000,000-fold. The combination of proteins in a cell confers specificity in the signals it detects and the responses it carries out. Scaffolding proteins increase signaling efficiency. Pathway branching further helps the cell coordinate signals and responses. Signal response can be terminated quickly because ligand binding is reversible. What mechanisms in the cell terminate its response to a signal and maintain its ability to respond to new signals? Concept 11.5 Apoptosis requires integration of multiple cell signaling pathways, pp. 229. 231. Apoptosis is a type of programmed cell death in which cell components are disposed of in an orderly fashion. Studies of the soilworm Sinorhabditis elegans clarified molecular details of the relevant signaling pathways. A death signal leads to activation of caspases and nucleases, the main enzymes involved in apoptosis. Several apoptotic signaling pathways with related proteins exist in the cells of humans and other mammals, triggered in different ways. Signals eliciting apoptosis can originate from outside or inside the cell. What is an explanation for the similarities between genes in yeasts, nematodes, and mammals that control apoptosis? Test your understanding. For more multiple choice questions, go to the practice test in the Mastering Biology e-text or study area, or go to goo.gl/gruwrg. Levels 1 to 2, Remembering/Understanding. Binding of a signaling molecule to which type of receptor leads directly to a change in the distribution of substances on opposite sides of the membrane. Intracellular receptor. G-protein coupled receptor. Phosphorylated receptor tyrosine kinase dimer. Ligand-gated ion channel. The activation of receptor tyrosine kinases is characterized by dimerization and phosphorylation. Dimerization and binding. A phosphorylation cascade. GTP hydrolysis. Lipid-soluble signaling molecules, such as aldosterone, cross the membranes of all cells but affect only target cells because only target cells retain the appropriate DNA segments. Intracellular receptors are present only in target cells. Only target cells have enzymes that break down aldosterone. Only in target cells is aldosterone able to initiate the phosphorylation cascade that turns genes on. Consider this pathway. Identify the second messenger. CAMP. G-protein. GTP. Adenylyl cyclase. Which of the following occurs during apoptosis? Lysis of the cell. Direct contact between signaling cells. Fragmentation of the DNA. Release of proteases outside the cell. Levels 3 to 4, Applying/Analyzing. Which observation suggested to Sutherland the involvement of a second messenger in epinephrine's effect on liver cells? Enzymatic activity was proportional to the amount of calcium added to a cell-free extract. Receptor studies indicated that epinephrine was a ligand. Glycogen breakdown was observed only when epinephrine was administered to intact cells. Glycogen breakdown was observed only when epinephrine and glycogen phosphorylase were mixed. Protein phosphorylation is commonly involved with which of the following? Ligand binding by receptor tyrosine kinases. Activation of G-protein coupled receptors. Activation of protein kinase molecules. Release of from the ER lumen. Levels 5 to 6, Evaluating/Creating. Draw the following apoptotic pathway, which operates in human immune cells. A death signal is received when a molecule called Fos binds its cell surface receptor. The binding of many Fos molecules to receptors causes receptor clustering. The intracellular regions of the receptors, when together, bind proteins called adapter proteins. These in turn bind to inactive molecules of caspase 8, which become activated and then activate caspase 3. Once activated, caspase 3 initiates apoptosis. Evolution connection. Identify the evolutionary mechanisms that might account for the origin and persistence of cell-to-cell signaling systems in prokaryotes. Scientific inquiry. Epinephrine initiates a signal transduction pathway that produces cyclic AMP, CAMP, and leads to the breakdown of glycogen to glucose, a major energy source for cells. But glycogen breakdown is only part of the fight or flight response that epinephrine brings about. The overall effect on the body includes an increase in heart rate and alertness, as well as a burst of energy. Given that caffeine blocks the activity of CAMP phosphodiesterase, propose a mechanism by which caffeine ingestion leads to heightened alertness and sleeplessness. Science, technology, and society. The aging process is thought to be initiated at the cellular level. Among the changes that can occur after a certain number of cell divisions is the loss of a cell's ability to respond to growth factors and other signals. Much research into aging is aimed at understanding such losses, with the ultimate goal of extending the human lifespan. Not everyone, however, agrees that this is a desirable goal. If life expectancy were greatly increased, discuss what might be the social and ecological consequences. Write about a theme, organization. The properties of life emerge at the biological level of the cell. The highly regulated process of apoptosis is not simply the destruction of a cell, it is also an emergent property. Write a short essay, about 100 to 150 words, that briefly explains the role of apoptosis in the development and proper functioning of an animal, and describe how this form of programmed cell death is a process that emerges from the orderly integration of signaling pathways. Synthesize your knowledge. A bowl of regular potato chips and a bowl of taco-flavored chips. There are 5 basic tastes: sour, salty, sweet, bitter, and umami. Salt is detected when the concentration of salt outside of a taste bud cell is higher than that inside of it, and ion channels allow the passive leakage of into the cell. The resulting change in membrane potential, c. Concept 7.4, sends the salty signal to the brain. Umami is a savory taste generated by glutamate, glutamic acid, found in monosodium glutamate, or message, which is used as a flavor enhancer in foods such as taco-flavored tortilla chips. The glutamate receptor is a GPCR, which, when bound, initiates a signaling pathway that ends with a cellular response, perceived by you as taste. If you eat a regular potato chip and then rinse your mouth, you will no longer taste salt. But if you eat a flavored tortilla chip and then rinse, the taste persists. Try it. Propose a possible explanation for this difference. For selected answers, see Appendix A.